Good evening, Dr. Saptak sir. This is Dr. Moshmi here from other end.
Uh,
Thank you, sir, for the presence. We will shortly start the meeting, sir, once the audience joined.
Hello,
पार्थ भाई, आवाज आ रही है?
madam, it's fine. I just want to check my slide once. I just want to share and check that it
Sure,
is
sure,
working on.
sure, Saptak sir. Please do that,
Okay,
please.
okay.
भाई, सप्तक सर को प्रेजेंटेशन राइट्स दे दीजिए ना, वो चेक करेंगे. Yes,
Madam,
sir. It is came. But just make it full slide.
okay. Is it visible now?
Once again. Yeah, perfect, sir. Perfect.
Okay, I'm just changing the slide. You just see that it is changing or not.
No problem, sir. We will.
So it's changing, right?
Not yet. Not yet.
Okay. Oh, it's. So.
No, don't stop it. Just
Okay.
click next slide. Don't stop it. Just click down arrow in the keyboard.
Yeah, I'm putting the down arrow, but it's not changing.
Or else, if in the mode, if you can see next slide option, that is also work.
Switch
In the
off.
mode of PPT, there will be a next slide, like where it is sharing in there, in that corner, you can, yes, 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 it is changing.
Okay.
Next slide, sir.
So it's changing?
Yes, sir. Cancer. Yeah, next slide.
Yes, sir.
Oncogene, tumor suppressor gene.
Okay,
Right.
so it's
Hallmark
changing.
of
Okay,
cancer.
okay.
Yes,
Okay,
yes.
it's from click, it's going on. Okay.
Yes, sir.
Okay,
Yes.
no problem. So I will stop it now.
Yes, yes, please, sir. Please stop it.
Okay. Thank
It's
you.
all
Thank you,
okay,
madam.
sir, from your side. Yeah.
Okay. I am audible, right?
Yes, you are perfectly audible, sir. No problem.
Okay, thank you. Thank you, madam. Thank you.
Yeah. Just a couple of minutes, sir.
Yes, yes, no issue.
Audience
No issue.
will join around 6 o'clock, so we can start then.
Okay, okay, okay. Thank you.
Yeah. Good evening, all the participants and dignitaries present in this fourth day of 6-day Atal FDP on recent therapeutic and diagnostic trends in healthcare and medical technology. Invoking the divine blessings of Lord Swaminarayan, Brahmaswarup Param Pujya Sri Sri Hari Prasad Swamiji Maharaj, Prakat Guru Param Pujya Sri Prem Swarup Ji Maharaj, and our beloved president and mentor of Atmiya University, Rajkot, Gujarat, Param Pujya Sri Tyagvallabh Ji Maharaj, I, Dr. Maushmi Das, along with my coordinator, Dr. Devashish Banerjee, welcome all of you in this 6 days Atal FDP program. And this is the fourth day. Successfully, we are running with the help of all the participants and the resource persons. Let's start the session with initiating Om chant 3 times. Let's close our eyes with folded hands. Take the blessings from that Almighty. Before starting the session, couple of announcements to my dear and respected participants. Today, we are going to share the attendance link in the Cisco WebEx chat box, as we do every day, after the start of second session, just after the finishing of the first session and before the start of the second session. It will be in the chat box of Cisco WebEx reflected, and we will at the same time announce in the WhatsApp group that we have already shared the attendance link in the Cisco WebEx chat box, and such reminder we will do 3 to 4 times in your WhatsApp group that we have shared the attendance of today in the Cisco WebEx link. So kindly be in touch with the Cisco WebEx link from the very start. The second thing is the feedback of today's session. We will share both in the Cisco WebEx chat and as well as in the WhatsApp group. For that also, we will share the same announcement when we were sharing that links. The third thing, couple of members are telling that they have yet not received the study material. Please don't worry. Your comfort is our utmost priority. So the participants who have yet not received the materials due to one of the other reason will definitely be getting the materials. Don't worry about it. The next thing is, after the session, we have a 1 or 2 question answer round. So you can put your questions in the Cisco WebEx chat box. We will take the questions. If anybody wanted to ask directly to the resource person the question, that is also there. The privilege is there. You can raise your hand. One option is there in the Cisco WebEx link, hand raise option. If you do that, our technical team will support you to unmute you and you can easily ask the questions. With this brief things, I request all the participants, please focus on today's first lecture. I take the opportunity with my coordinator, Dr. Devashish Banerjee, to introduce our first speaker of the day, a very commendable person with a very vast experience, Dr. Shaptak Banerjee, senior scientist, officer, grade 2 at Department of Immunoregulation and Immunodiagnostic, in Chittaranjan National Cancer Institute, Kolkata. Mr. Banerjee has a very great experience in the research endeavors in area of oncology and cancer. Being a PhD in 2014, the journey of this person has progressed through several milestones of the achieving great credentials, being postdoc in very reputed institutes of India, namely the National Institute of Immunology, New Delhi, under DBT funded project, National Institute of Research in Tuberculosis, Chennai, MRC DBT project, beside a NET, CSIR NET JRF. He is also bestowed with various prestigious memberships of government and at both national and international level. Worth mentioning few are Indian Association for Cancer Research, American Association of Indian Scientists in Cancer Research, European Association for Cancer Research, American Society for Clinical Oncology. He is also a reviewer in Science Engineering Research Board, ACRB. A great number of publications in book chapters, book. He also had in his credentials with a worth mentioning H index and I10 index. His highest impact factor till now, what we have in our search repository is 15 of impact factor in cell death journal. With this glimpse of his brilliant career, I, with my coordinator, Dr. Devashish Banerjee, would like to take the opportunity to invite Dr. Banerjee in this particular platform. I am sure you participants will definitely get today few new glimpses on the area of cancer and oncology from his expertise and from his deliverables. Thank you so much. Dr. Banerjee, I would like to request you, please take the floor. All is yours. You have been provided presentation rights. Please share your presentation.
Good evening to everyone. Thank you, Dr. Moshumi, and thank you, Dr. Devashish, for giving me the opportunity to present my work and some of the basics of cancer immunology I will discuss today. The area I am working. So is my slide is visible?
Yes, yes.
Okay,
It is.
okay,
Yeah,
it is
please
in
go
full
on.
screen mode.
Yes,
Yes.
sir.
Okay, okay. So I'm Dr. Saptagunanji from Chittaranjan, Senior Scientific Officer from Chittaranjan National Cancer Institute, working in the Department of Immunoregulation and Immunodiagnostic. Today I'm presenting my topic is novel approaches to restrain breast cancer by targeting metabolic and immune landscape. We all know that cancer is a dreadful disease and it is having different so I will just give you a basic introduction, maybe someone who is not familiar with the thing, that cancer is not a single disease. It's a mixture of complex disease, and it doesn't happen in one day. It's a step-by-step process which takes quite a long time. So here you can see that if you see, if my cursor is visible, so this is the normal mucosa where you can see different type of cells are there. And then here from there, it's like hyperplasia, dysplasia, carcinoma in situ, and invasive cancer. So where it happens, like in initial stage when there is some cells are getting mutated, they start to lose their structure and function. And slowly you can see that they are missing. And finally, like carcinoma in situ, still now we can treat the cancer very well because it's not spread anyone. The margin is still there. And once it's breaking out, then it's like malignant cancer or carcinoma. And depending on the tissue from which it's arising, we can differentiate it into carcinoma, like if it is from epithelial cell, sarcoma when it's from bone or soft tissue, leukemia from WBC, lymphoma from B and T cell, multiple myeloma when it's from plasma cells, and melanoma when it's from the cells which are giving our skin color, that is melanin synthesizing cells. So broadly, and depending on the organ, we call it breast cancer, lung cancer, but actually what type of cell it is, that is the most important. So most of the cancer are mainly carcinoma. If it is in breast, it's breast carcinoma. If it's in lung, it's lung carcinoma. So we name it like that. Next, coming to the few genes, like there, we can broadly classify them into two groups. One is proto-oncogene, another is tumor suppressor gene. So proto-oncogenes are the genes which, if they are staying normally, there is no problem. They are doing their function. But once they get mutated, they gain some extra function. So that is gain of function. And then they became cancerous. Like example, like HER2, RAS, MYC, if they get mutated, then they become oncogene. So till now it is proto-oncogene. Once they are mutated, they become oncogene. Another type of genes are there, which are also called the guardian of the genome, like tumor suppressor gene. Their work is to check the cell cycle that all during division of cell, all DNA is intact or not. If there any problem, they stop it and give the time, they interrupt the cell cycle, give the time to cell to repair it. If it is properly repaired, then it is allowed to the next stage. And if this control is lost, so in this case, it is loss of function, then the cancer arrives. So P53 is the main gene who is responsible for controlling our whole genome. That's why almost 80 to 85% cancer, we can see that P53 is mutated. So next, coming to the hallmarks of cancer. So this is one very famous paper from Douglas and Hanahan and Robert Weinberg. In 2000, they first came out with this, where they find out some salient features which are called the hallmarks of cancer. So if these conditions are there, we can tell that this is a cancer, like evading growth suppressor, like you cannot stop the growth. Then like activating invasion and metastasis. I was just telling you that until they do the invasion to the nearby tissue, margins are break, it's in benign stage. Then like reproductive immortality. So they don't need any signal and they keep continuing dividing. Induce angiogenesis, that is formation of new blood vessel. I will describe this later. Then resisting cell death. So like we know that normal cells have certain lifetimes, after that they die. But in cancer condition, there is no cell death like that, and continuously they are getting proliferation, sustained proliferating signal. So normally, if we see any cell cycle, any signaling, like it's there is a intercellular and extracellular domain. When the condition is favorable, the signal transduces and cell divides. Once it is not required, it stops. But these type of controls are lost in case of cancer. So next, in 2011, they came out with some more addition, that is next gen hallmarks of cancer next generation in 2011 by these two famous person. You can see the citation also. So it's like very prominent paper when every one of us has to start with, because these are the thing where everyone is working. So here they added avoiding immune destruction. Like our immune cells are very important for keeping the any disease or cancer under control. Once that is deregulated, then only the cancer can take over the thing. Then like tumor promoting inflammation, genome instability, mutation, and deregulation of cellular energetics. Again, today I will discuss some of these parts too. What happened? It's not moving. Yes. So till now we can see that this many type of markers we can find, like these are the hallmarks. And in 2022, they came up with some new model. So like after every 10 to 11 years, they are adding some new features. So it's like epigenetic reprogramming, polymorphic microbiome, because now microbiota is also playing very important role in cancer. As we know, Helicobacter pylori is very famous. like for this pancreatic cancer, cell senescence, and phenotypic plasticity, like cell can transform from one form to another. So these are the salient features till now we can get in 2022, and today I will be focusing on mainly, like you can see many parts are there. I will be just trying to catch up these three, like cellular metabolism, immune destruction, and cell plasticity and angiogenesis. So what is tumor or tumor microenvironment? Generally, in your mind, it may come that tumor means it's having only the tumor cell, which is not true. It's composed of tumor cells along with different type of immune cells. There are other cells like endothelial cell, fibroblast, and pericytes which supply the blood to the tumor. There are different neighboring cells are there, which are like associated cells. So today we'll see how they interact with each other. So this is how it looks. It's very chaotic if you see. So here you can see that within the tumor there are some necrotic zone. So it's black marked. So you can see there are some places where oxygen level is very low, because these are the peripheral region where the blood supplies are only there, and immune cells also can enter till then. After that, there is no communication between immune cell or drug that cannot go, and inner core become very high in pH, so immune cells even they become dysfunction. So this is how broadly a cancer, if you cut it, it looks like this is the main blood vessel supplying blood within the tumor. And as soon as the peripheral regions are getting the blood supply and oxygen, they proliferate so fast that inner core always stay hypoxic. So I have to talk about this person, Dr. Judah Folkman, who is also famous as the father of angiogenesis, because he was the first person to find out that angiogenesis is one main cause without which a tumor cannot become malignant. He also find out the receptors like VGF, different vascular endothelial growth factor receptors, their vascular endothelial growth factor, and their inhibitor to grab this angiogenesis. So in our body, it is the system is made in such a way that blood from blood, oxygen, we know oxygen nutrient, they just goes to the neighboring tissues. So all these cases, the distance is maintained such a way that it is less than 100 micron, because oxygen and all can only go till that much. So that's why it's another way our cellular architecture, like tissue architecture, is controlled. So if you see a cut has taken place in your body anywhere, slowly from all the sides it's come up. Once they meet to each other, they cells stop dividing. They just don't keep on dividing. In cancer condition, all these controls are lost. And this is how typically a tumor will look around. If you see its vasculature and its blood, there are some spaces which are highly necrotic, so they don't have any supply of oxygen and all. So there, after that, there are some hypoxic zone where it is moderate, and these are the places where the stem cells, cancer stem cells, stays, and they are the most resistant type of cells, which are mainly responsible for all type of tumor heterogeneity. I will discuss later. So this is what I was telling. This is, if this is the blood vessel and you can see the oxygen level, this is normoxial. So up to this much, all immune cell or drugs even can go up. And these are the cells which are the very fast proliferating cell. After that, there is a hypoxic region where mostly the resistant type of cells or cancer stem cell stays, inner core is always necrotic. So these cells never get adequate amount of oxygen or nutrient. So they continuously keeps giving a signal that we are not getting enough food or oxygen. And as cancer cells are also belong to us. like it's not from foreign particle, like any bacteria or virus which is coming from outside. Body cannot recognize this signal, and they think that some of our own body part which need to be given oxygen is not getting it. So they starts giving more supply towards the tumor, and tumor proliferates faster. Here you can see this HIF1 alpha, that is hypoxia inducing factor 1 alpha. This is very important protein which takes place in this cancer angiogenesis process. So in normal condition, like when the normoxia is there, that the oxygen level is perfect, then the mRNA itself is get degraded. If the protein is prepared, protein is made, that is ubiquinolated and then degraded. But when there is hypoxia, then this HIF1 alpha is not degraded, and that translocate within the nucleus, and that sits with along with HIF1 alpha beta, CBPP 300 and other cofactors, and slowly they starts producing all required genes which are required for cancer progression. That is VGF, FGF, all these type of growth promoting factors for cancer, they keep supplying. Again, this is very basic. I am just touching up, that is VEGF receptor 1, this is VEGF receptor 2 and receptor 3. These are neurophilin 1 and 2. So mainly VEGF R1 and R2 are responsible for angiogenesis. VEGF R3 and NRP, NP1s are responsible for lymph angiogenesis, which today I am not touching up. And among these, the VEGF R2 has the highest potential to perform angiogenesis and cancer progression, whereas VEGF-R1 acts as a decoy receptor. So if a VEGF-A is binding to either VEGF-R1 or R2, in case of R1, the signaling will be very less, so the effect of angiogenesis will be less. Whereas in this case, the signaling is 100 times more, so that is very aggressive. That's why most of the time we are trying to block these VEGF-R2 receptors in cancer. So this is the angiogenesis process. You can see that this is the lumen, this is the inside the blood vessel. So if a tumor starts growing, until and unless it is invading, we can see it's in situ, and it can be a benign tumor or maybe a tumor in the process of becoming malignant. So they secretes different angiogenic molecules. Normally, our endothelial cells, like the main cells which are lining the blood vessel, they don't divide. But once they get this vascular endothelial growth factor, it's a signal for them to proliferate. And slowly the sprouting starts. These are called the tip cells, endothelial cells, and the matrix has to be broken down. So different MMPs works here to break down the matrix. More signal comes to this, and slowly there is a sprouting starts going towards the tumor. Once it reached, then one way, like tumor gets the more oxygen and nutrient, and the same time some cells can dislodge within this, and they are like metastasizing tumor cells which can spread throughout the body. So if we can detect it early, we can stop it before this place. It's very easy to control cancer. But once this stage is crossed, then it's very difficult. This is how a normal blood vessel looks like. These are endothelial cells. These green colored cells are pericytes, which are covering the endothelial cell. Over that smooth muscle actin are there. But once in cancer condition, you will see this regulation is lost. So like it's very chaotic, torturous. So neither immune cell can go inside or drug can go inside and do their function. So they are leak out in most of the cases. So that's why like tumor angiogenesis is also linked with metastasis and tumor progression. So this is again the same way. We even up to this stage we couldn't understand that what's going on. So most of the time these are undetectable. Once it crossed, then only it starts rapid growth, and by the time the patient can understand there is some stigma, so the initial stage they don't come. Once it comes to the advanced stage, like it's very difficult to control. Breast cancer stem cells are very important, like any cancer stem cells are very important, play very important role in this condition. Like they also from here they makes the endothelial cells and make helps in sprouting. In some case you will see vascular mimicry. So here these are the endothelial cell, but here is there some gap. Cancer still lines themselves in such a way that seeing like with normal using hematoxylin staining, they looks like a blood vessel. But if you go for the marker study, you will see they are negative. So that is called vascular mimicry. They just arrange themselves in such a way that they stops the immune cell to go inside. At the same time, they makes a false blood vessel so that they can get the food or oxygen or nutrient. So this is a real type photo from where like from one of my publication where you can see that how it looks. So this is the main blood vessel. From here you will see lot of blood vessels are coming. So we in my PhD my work was on tumor angiogenesis. So there we used to treat with some drugs and see how this is coming out. Again, this is like B16 melanoma, this is a leach carcinoma, two tumor model. Here you can see if the tumor is small, angiogenesis is more less, the tumor is small. But with bigger tumor and metastasis, you can see their thick blood vessels are there. And here also like histologically, you can see these are when it's under control and these are like big torturous thing. CD31 is marker of endothelial cells. So if we want to study the angiogenesis, like this is with Duffy stain, like nuclear stain, we can see that normal angiogenesis and cancer angiogenesis. Here also, like you can see, this is the big tortuous blood vessels are there. And these are the pericytes which should be sitting over the endothelial cell, but they are disorganized. Where if it is it could be bring back to the normal condition. You can see here that over the endothelial cell, that NG2 cells are sitting and the blood vessel, it is not thick, it's not tortuous. So these are called vascular normalization. Again, here you can see the expression of VEGF is very high in control group. This was a drug from our lab we were working that time, which can bring. So this is for just for the example I am showing you. Here another photograph you can see where the EC is the endothelial cell and P is the pericyte. So here the endothelial cells are disoriented and pericytes are sitting elsewhere. Where in normal case you can see that these are the endothelial cells. Here these are the pericytes, big cells which are covering it. So blood vessels become in normal condition. So next I am moving on to the metabolism. So tumor cell metabolism is very unique from our normal body metabolism. We know that most of the time we do the aerobic oxygenation, aerobic glycolytic pathway. So from a glucose, if it goes inside, from glycolysis, it goes to pyruvate. Pyruvate goes inside the mitochondria, OXPHOS pathway. And total, we used to get 36 ATP from here and 2 from anaerobic. So it's 38 ATP. Whereas in cancer condition, it's always only the glycolytic pathway. They never goes the mitochondrial OXPHOS pathway. So in that way, you can see it is not very energy efficient. It can see from one glucose, it gets only 2 molecule of ATP. The another thing is that in our body, like when we are doing exercise or running, only that time our normal muscle cell they goes this anaerobic oxygenation. That's why the lactate is formed and we get fatigue. But after some time that goes off. In cancer condition also, like this phenomena was discovered by Otto von Warburg and named after him. That's Warburg effect. Even in the presence of oxygen, they always take the glycolytic pathway and never take the mitochondrial pathway. So what is the advantage? I will just tell you. So this is the glycolysis pathway we know. From each step, they are mainly focusing on that they has to grow rapidly. So for rapid growth, they need the cell membrane. and they also need the different nucleotides like ATGC, this purine and pyrimidine. So from each byproduct, you can see they makes the NADPH from where they get their actual ATP. And from there, they are getting all different types of things like for biosynthesis for complex lipids because it is required for their cell membrane, then folate cycle, methionine cycle, all the things which are required for their fast proliferation, they synthesized it. And finally, the lactate, which is formed, that is also given to the nearby cells where even this glucose is not reaching. So lactose is utilized by the nearby cells for producing production of ATP. And this high lactate also dysfunctions the immune cells and give them another sort of protection. And as they are not taking that oxidative phosphorylation, so in cancer cells, the generation of ROS is also minimized. In that way also, they are protected from the cell death. So this Warburg effect is very unique phenomenon which we can target to differentiate between normal cell and cancer cell. So next coming to the 3 E's of cancer. So these are called elimination, equilibrium and escape. So in normal condition, our immune cells are competent enough to find any tumor cell and kill it. So if it is killing the tumor cell, so we are getting the protection, so it is elimination. At some stage, when there is a balance, like if 10 cells are killed, 10 cells are formed, so it's a static condition. So that is equilibrium. And once that also breaks, like immune cell completely fails to eliminate the tumor due to continuous mutation within that cancer cell, then it's a escape. So till now I was telling you different salient features of cancer, how they are different. So now from these are the real-time photograph from where you can see how a cancer cell looks under microscope when compared to a normal cell. In cancer cell, you can see the nucleus is very high, very big, very dense, whereas this is in normal condition. Because their main function is to divide rapidly. Sometimes there you will get like very less amount of cytoplasm along with them. Because their main aim is to outgrow and grow rapidly as fast as possible. And everyone is different from each other, you can see. There is no similarity. The size and shape is also different. And so all normal things are lost between cancer and normal cell. So there is also some difference between normal stem cell and cancer stem cell, where in this case they also have the self-renewal capacity, but in this case it's infinite, like it never stops and it never dies. It's like that. So normally it forms an organ, and in this case it forms a tumor. It is highly regulated self-renewal and differentiation, but in cancerous condition, it is highly dysregulated. So these are the few comparison. If you want, you can keep it. So now I am coming to my main topic, that is breast cancer. Why I have chosen to decided to work with breast cancer. This is the Globocan data from where you can see the number of cancer cases in breast cancer is rising is the highest. This is for males and this is for females. These are the top cancers within India. I have put India. So in Globocan, if you put the country and different condition, you can set they will give you the result. And in females, the breast cancer is the highest. You can see it's almost 26.6%. And mortality rate also it is highest. Though lip and oral cancer is highest in male, but when compared to the mortality of both sexes, breast cancer again tops because of the recurrence and tumor and treatment resistance. These are the main problems with the breast cancer. And this is not only the problem of India, this is the scenario of the world. You can see all over the world. If you put like breast cancer here, you can select the age groups, how we will put, it will change accordingly. So I have just given 0 to 80. So it's like almost the whole world is affected with breast cancer. And again, breast cancer is number one. New cases for death and 5-year prevalence, like recurrence. In all cases, breast cancer is on top. So now coming to the types. So the again tissue from which the cancer arises, like this is the, if it is from duct, we call it ductal breast carcinoma. If it is from the lobules, then it's lobular carcinoma. If it is mixed from both the cases, it's mixed. This is very rare condition like inflammatory breast cancer, and this is mucinous. They form say thick mucin, and stages are dependent on the size of the tumor and with how many lymph node it has transmitted, metastasized. So depending on number of lymph nodes and size, there are certain guidelines which are getting updated every year in different conferences. They select it. So I am not going deep into that, but I am just giving the basic how we find the stage types and all. And this is the basic treatment modality, where if it is localized like in situ condition, when there is no invasion has taken place, doctors generally go for surgery, and residual body can be treated with hormonal therapy or radiotherapy depending on the patient profile. If it is in early stage, then also they goes for surgery followed by chemotherapy and radiotherapy, and depending on different hormonal therapies also there. But if it is in advanced stage, generally most of the time we get patient in advanced stages. So they first reduce the tumor, giving some chemotherapeutic drugs, and residual body is removed by surgery. So now coming to the breast cancer stem cells, as these are the main cause of the tumor heterogeneity and recurrence. So like this is the memory stem cell from where it goes to luminal stem cell and basal stem cell. If any mutation taking place within this, that can become a breast cancer stem cell. If the progenitor, like ductal progenitor, alveolar progenitor, if epithelial, myoepithelial, from anywhere, if it is mutated, that also can be goes to breast cancer stem cell, or even after forming the final stage, that also can get some mutation and transformed into tumor cell. So these are like unknown areas. These are the gray area. That's why it's all discontinued lines are there. Now I'm showing you that why these are, every time I'm telling them that they are very important. So if you see, this is a tumor and this is the anti-cancer drug is given. These are the fast growing proliferating cells where the drug can reach. They eliminate this. And I told you there are certain pockets where neither immune cell nor drug can reach. Hypoxic region, there the stem cell or residual disease stays, and most of the time the tumor stem cell stays in that condition. So if this is the remaining body, from here again that they can make the whole tumor. So that's called the relapse. This cancer stem cell has the capacity of two-way replication. One is symmetric, like normal cells how from mother to daughter. So in that way, they from one stem cell, they can produce two stem cells. And there is a asymmetric division where they can give rise to different types of cancer cells. So with like different mutations. And so that's why it's very difficult to target, because by the time immune cell find and kills one type of cells, another cell type, they are getting mutated to B, C, D like that. So it's very difficult for the immune cell to find and kill, or it is also difficult for us to target because it is not one type of cell, it's having different types of cells. This is a remarkable paper from Muhammad Al-Haji, who was first to find out that within the tumor there is a specialized group of cells, and he found out this is like CD44 positive, 24 negative to low lineage negative cell, which can even as few as 100 cells can give rise to tumor. So I'll shortly discuss this paper once, like in mice tumor, if you give like high number of cells, like this is 8 mice they have given unsorted, like whole tumor, all 8 took up the tumor, and slowly they decrease the number of cells. It is only 10 to the power 5, it's 5 into 10 to the power 4 like that. But when it's 10 to the power 4, only 3 out of 12 mice got tumor, and there was no tumor in these mice. When he has sorted, like they have selected out from the whole tumor CD44 and CD24 positive cells. Yes, like 2 days back, Dr. Ratnadeep was telling you about the flow cytometry. So I was listening that I'm not discussing that. So I'm directly telling the markers. So when they sorted this and given to the mice, there was no tumor was found. But when it is CD44 positive, 24 low or negative, you can see 10 out of 10, even at the level of 10 to the power 3 cell, it is giving rise to tumor. Then they search another marker like ESA positive and negative. Depending on that, like ESA positive cells, you can see like if 100 cells are given, which are 44 positive, 24 negative, or low ESA positive, that can give rise to a tumor. In 200, it's 4 out of 4, 500, 4 out of 4. So that's why this, from this paper only, this idea of cancer stem cell came, and then we are now searching different markers for different cancer stem cell. Like for lung cancer, it's different. For oral cancer, it's different. So it opened up a new horizon of work. And this is from patient sample also. Like they are also, they have found out like that. And we are also following these same markers for our work. I will come to that later. So this is the ESA positive 24 positive cells. They have taken ESA positive 24 negative. So this is the cancer stem cell. This is again like 24 negative, 44 positive. These are cancer stem cells. So if you anyone interested, they can read the paper. And this is the, like in mice they have given where 24 negative cells are there. You can see the big tumor is formed with the histology, and here very small tumor is there, because if cancer generating cells are not there, these cells will die out and cancer will go off. But main problem is this breast cancer stem cell, because from there the whole tumor is coming there. That's why we are trying to target this breast cancer stem cell. So when I started my journey in Chittaranjan National Cancer Institute, I after getting all the information, I wrote this. We came up with this paper where you will see cancer stem cells and immune cells interaction within breast cancer. So here you will get like all immune cell, how they react, how they interact with cancer cell. So, like cancer stem cells are responsible for EMT metastasis, chemoresistance. Their cell cycle is very slow. They go asymmetric division, tumor sphere formation, and they are responsible for the tumorigenicity. And like here, you can say that memory epithelial cell, from there, if the mutation they acquire, it can turn out to be a cancer stem, breast cancer stem cell, or from progenitor, or from any other differentiated cell, it can come. And here, that time, the information how much we had, it was like they interact with gamma delta T cell, but its markers are not much known. CD8 like PD, PDL1 is there. CD4 helper cells are there. Again, like Ratnadeep that described this heterogeneity of T cells. So I'm not going, I'm directly going to the markers. And these are the regulatory cells, which are very, very important in case of cancer. And this is another part where the breast cancer stem cell was also interacting with platelets. In my last part of talk, I will discuss this, that how they are interacting. So they are also dysfunctioning the NK cells, so they lose their cancer killing property. So now coming to the my work, what I am getting now, I am doing this. Like we are working with 2DG. So this is a very cheap drug way, you can say, and with very less or no toxicity, because it's a analog of glucose. only this weight instead of its age. So it's deoxyglucose. So how it works, like normal glucose, we know it after hexokinase goes to glucose 6 phosphate and so on. But this 2-deoxyglucose 6 phosphate acts as an inhibitor for further pathways. So the glycolysis path can be targeted very easily with this glucose 2-deoxyglucose. And if this, so the more they will take, the cell will take, the more 2-deoxy 6 phosphate will be formed. With feedback loop, it can also block the hexokinase, and the cell will be deprived from the generating ATP. So again, I am coming to this photo. I told you during Warburg effect that if we give one 2DG and one glucose, like two molecules we are adding together to a normal cell, from normal glucose, normal cell will make 36 plus 2, 38 ATP, whereas cancer cell will make only 2 ATP because it don't take the Oxford pathway. And from 2DG, no one will able to get anything. So we can directly target the cancer cells, especially breast cancer stem cells, in that way because they will be deprived of ATP and their other thing. Because normal cell will only divide when it is required, and anyhow it is getting 38 ATP from one glucose, and it is getting only 2 ATP from one glucose. So in this way, we can selectively target cancer cells using this 2DG. And this is also very cheap because we know the cancer treatment is very costly when we are talking about any monoclonal antibody or any type of therapy. So this 2DG is very cheap because it's just analog of glucose and with very less or no toxicity. So what we are doing, we have taken the supply, the breast cancer can be defined as luminal A, luminal B, HER2 and triple negative depending on the presence of different hormonal receptors, that is estrogen, progesterone and HER2. So in this case, all the receptors are present. So we can target them with any type of receptor like hormonal therapy, and it works very well there. And the progress, like cancer progression, is very good. Like we can control it in very nice way. It is very much problematic when it comes to HER2 or triple negative, because in triple negative all are negative, so no hormonal therapy works here. Here some trastuzumab or some targeted drug which can target HER2 can be used. So in this in my lab I am using 2DG to target all these type of four subtypes. We have differentiated them in four subtypes, and we are studying their effect on cancer stem cell. So these are the few reports available when we started. That 2-deoxyglucose inhibit aggressive triple negative breast cancer. They also enhance T cell memory cell action. So no adverse, not much adverse thing was there for 2DG, and so we started working with this. So this is the protocol, like do it. So we know from the doctor that what type of cancer it is, luminal A, luminal B, HER2 or TNBC. We mechanically dissociate them. Then we used to treat them with collagenase, make single cell, and then we centrifuge it. Then we do max sorting. This is magnetic sorting, where we use CD44 antibody tagged with magnetic beads, and here the strong magnets is there. So after some time, the 44 positive cell will be stuck there. We used to take out the rest of the thing, then go for CD44, 24. So there 24 positive cells will be stuck, and we used to take out the 24 negative cells. Like this, few steps, we purify the cancer stem cells from patient sample, and then we culture them in our culture plates. These are just to differentiate four colors we have given it, nothing else. We treat with 2DG, and the primary tumor is formed, mammosphere. From there, like it's 3D tumor, and from there we go, these tumor are dissociated, and we go for the secondary mammosphere formation, and followed by in every step we used to check their transcription factors, and markers using flow cytometry and PCR, Western blot, all other things. So this is unpublished data. So I'm just sharing with you. So this is the primary mammosphere, how it looks. It is for luminal A, and this is for luminal B, HER2 positive TNBC. In secondary, when we enrich them, it's like called like only cancer stem cells will be proliferating. Other cancer cell will not proliferate and they will die out with time. The media is like that. They only helps to grow the cancer stem cell. It's stem cell media. And we are also sorting the cancer stem cell prior to this stage. And after 2DG treatment, you can see the mammosphere size is much smaller, and we can like use the 2DG for that. Here also we have checked the status of CD44, SOX2, NANOG, OX4. These are very important transcription factors for these cancer stem cells. And every time you can see the NANOG is very high. So if after treatment we can bring down the NANOG, then we can target more specifically to these cancer stem cells. And our lab is mainly focusing in this area where we are studying the interaction of breast cancer stem cells with different type of immune cells. And we are using different therapeutic targets to kill the tumors. So here you can see this is luminal A, this is luminal B, HER2 and TNBC. So this is controlled like when no treatment is there, the cancer stem cell proportion is 2.4, which is 3.7 in luminal B, 8.29 in HER2 and 10% in TNBC. So this is another thing, as the high number of cancer stem cell, the TNBC is more aggressive and more difficult to target. And within this cancer stem cell, when we have checked the status of OX4 and NANOG, you can see the NANOG is very high, like 7, here it is 9, 17, and 26. And after treating with 2DG, we can bring down it to certain stage. And this is one of the reasons the cancer mammospheres are slowing down in growth and they are like many cells are dying. So next, what we do, we take the immune cells, specifically CD8 cells and the stem cells from the same tumor, and we co-culture them to create an environment that if they were, this could be happen within the tumor, how they are reacting to each other, and we study their functionality. So here you can say that till now the literature which is there, like cancer, like in the stem cells, they interact with all types of immune cells in different way. So this is for general CSC. I am talking about breast cancer stem cell. So I'm giving few. I'm telling few markers about the T cell functionality before going to my result, like activated T cells. What are they like? When the dendritic cell or antigen presenting cell encounter with any tumor, they kill it, they bring specific markers or molecules which can be recognized by T cell and they get activated and go and kill the tumor cells which are having the same thing. So the cytotoxicity is very high for them. They have high CD8, CD28, OX40, IFN gamma, TNF alpha, everything is very high, IL2 is high. Next, coming to the energy, T cell energy. So when it is suboptimal level, like in cancer cell, it's called epitope shredding. They don't first of all, they are our own cells. So maximum proteins are same. It's very difficult to distinguish between cancerous targets and normal cells. But the few antigens even which are there, they keep it in such a low condition that T cell don't get activated adequately. So this is called energy T cell. So in that cases, you will find IFN gamma level is low, 10 is high, that is IL2 is low, PD1, CTLA4 are high. And next comes the exhaustion. Here we will see lag3, TIM3, PD1, all are high, IFN gamma 2 are very low. And next is coming to senescence, like the cells are going to die, like repeatedly these cells are stimulated and finally they are dying. So 27, 28 is going down, and these are KIR, killer-like receptor, they are high. So in this way, like T cells can be, is not one population, it's very, very dynamic, and they can interchange between themselves. So again, another slide I am putting for this, like with aging, the telomerase length, we know it goes down. So that's why they goes down, it's a senescence. Sometimes they see it, but I showed you there are like lot of matrix are there who are stopping the cancer immune cell to go inside the cancer and kill it. So after some time when they can't go, they just ignore it. They see the tumor cell, but they don't take. So it's called ignorance. Then it's energy, like dendritic cell not able to activate the T cell, affected T cell effectively. Then it's exhaustion, like many markers are there, and famous, most of famous are CTLA4 and PD1 because we got Nobel Prize for that, like Hanzo and this person, they got Nobel Prize for this. And this effector cells, like it is apoptosis, once it's dying, it cannot, these Treg cells, they kill the effector T cell. So in that way, there are a lot of interplay taking place within the tumor microenvironment. Like CTLs are good for cancer killing, but they are inhibited by other function. So they cannot get activated properly. So now coming back to the my previous slide. So here we have seen, this is in case of luminal A, luminal B, R2 and TNBC, four subtypes. I am studying for PD1. This is the basic level of PD1, like T cell expression, like 8%. When it goes to the luminal B, it's 6. In HER2, it's 10. Its TNBC is high. So one way we saw that in cancer, stem cells are high in TNBC. Here you can see the PD1 positive T cells are also high in TNBC. So more cancer progression is taking place. The T cells, which are mainly affected T cells, are getting dysfunctional. So they cannot kill the tumor. And when we are co-culturing them with cancer stem cell, the PD1 expression goes up from 8 to 10, 6 to 11, it's like that. Same thing you can see in KLRG1. And if we treat with 2DG, that can bring it down to somehow the normal condition. For this is for luminal A, this is for luminal B, this is HER2, this is TN, this is like so high, we cannot, like once it is up to after some limit, it's very difficult. So we are now studying the molecular signaling, that what is the interrelation between this cancer stem cell and this PD1 KLRG positive high T cell. How they are, is there any way they are controlling each other? So currently we are studying this, and manuscript is under submission, so I cannot go details into that mechanism. Once maybe next time, once it's published, I can share it to you. At the same time, you can see this is the four subtypes. The IFN gamma and granzyme, which are the marker for affected T cell, these are much better in luminal A. So the progressive cancer progression is less here. We can control luminal way much better way, which falls drastically in case of TNB. Both the things. So cancer killing efficacy. So the cells which are responsible to kill the cancer are in very bad condition. That's why immune cell evasion, as I told you, the immune landscape, we are studying the immune landscape in different subtypes, molecular subtypes. So one way cancer stem cell are high, another way that immune cells are dysregulated, so they cannot do their function. Even with co-culture of stem cell, you can see it becomes worse. So next we check what is the effect of 2DG on healthy blood. So here you can see that there is not much difference between untreated and treated group. And this is how it looks in culture. There is not much difference. So 2DG is not doing much harm to the T cells. So from here we can come to the conclusion that NANOG and TNBC, like this is the one Reason maybe TNBC is so difficult to treat, cancer stem cell proportions are high, and 2DG can be a very good candidate to target in cancer therapy. So I am coming to the last part of my presentation today, where I will discuss the interaction between platelet and breast cancer stem cell. So as we know that invasion and metastasis is a hallmark of cancer, there are few publications. We know platelet for blood clotting for years. That platelets are required for our blood clotting. So this is a that they only do this work, but they are also playing very important role within cancer. So these are few papers you can see the tumor educated platelet for breast cancer detection. So they are also helping metastasis. So how they are doing? These platelets in cancer condition transform and they becomes activated platelet with different molecular markers, and they cover up the tumor and take it from one place to the secondary site. So this is how it looks actually. This is cancer cell and these are platelet in normal condition. In cancerous condition, they get activated, they sit and covers the whole cancer cell and they take it to the bloodstream. So when any NK cell or T cell interact to kill it, they can see that this is only a platelet plaque which is flowing in the bloodstream. So they do no harm to this cancer cell, and this is where they are evading the immune destruction. So this is the like tumor growth, immune escape, and extravasation. They also helps them to form the new niche because these platelets don't have nucleus, but they carry certain mRNA along with them, and they secretes the pro-inflammatory side, growth factors like VGF, PDDF, which are very helpful to form the new niche to the cancer. So another view you can see that this is the primary site. From here, if it has to come to the bloodstream, the angiogenesis has to take place. So I already described that how it is taking place. So once the tumor reaches here, so all the matrix has to be broken down. There is to be sprouting. So once the angiogenesis takes place, the cancer cell that can come into the circulation. But in our blood circulation, the blood pressure is so high that cell will die. But platelets, again giving them a coverage like this. They cover the whole thing like few cancer cells, they dislodge by EMT. So like endothelial to epithelial to mesenchymal transition. So here you will see the cell is big in structure, it become linear. So there is a structural change. It is called EMT, another hallmark of cancer. And then it's covered up by the platelets and carried through the bloodstream, and they go to the secondary site where these platelets release the required thing which are necessary for the cancer to form the new niche. So in this way, these platelets play as a chameleon in cancer. So there are many integrins, P-selectins, which are very important and crucial for this binding, especially this P-selectin. So in my another project, I am working with another student. So she is studying this interaction like cancer stem cell and activated platelet. And again, I will only focus on EMT and metastasis. So this is the way how we separate the platelets. Like in acidity, we collect the blood, then we centrifuge it, take the platelet-rich plasma. We go for second centrifugation where we take the platelet pellet and platelet poor plasma is discarded. Pellet is washed with Tidates buffer. Then when we need this aspirin, another molecule, like this is also very cheap. All we know that aspirin can be used for headache or something, and due to some reason it was stopped. But currently, we have found out the role of aspirin in cancer treatment. So they can keep the activated platelet in normal condition, like resting platelet condition. So this is how it looks under scanning electron microscope. This is the normal platelet, and this is in luminal A, and this is in TNBC. It's highly disorganized, like many platelet forms, making a big clump. Here, using flow cytometry assay, we can see that CD41 is the main marker of platelet, and P-selectin is the marker of activated platelet. So in normal peripheral blood, you will see P-selectin positive, 41 positive in 6.8. But when it's luminal A, you will see this proportion increases. In TNBC, it is much higher. And we have also seen, I'm not showing it here because again it's under review, the paper is submitted under review, where we have found that higher platelet numbers are associated with higher cancer progression. And we can see that aspirin can bring down this activation level. This is unstained like control. This is TEP, like uh, tumor educated platelet, where it P-selectin level was 18, which can be bring down to 9 using aspirin. And this is for luminal. In luminal cases, the activation level is very high. So again, we are trying to find out that how these activated platelets are interacting with breast cancer stem cell, and how we can abrogate their interaction so that they don't go for aggressive form, or is there any special marker we can find out which can be used as a biomarker for cancer metastasis. So similarly, we from patient tumor, we make the single cell of tumor. With that, we co-culture the resting platelet, like which is having aspirin, and activated platelet, which is not having aspirin, and then we sort the stem cells. So I just showed it in my schematic that we do max. This is how it actually looks. These are the tubes. Here, if you see, very strong magnet is there, and these antibodies are tagged with the markers one by one. We has to sort them out. Can you see a black streak in this case? If you can find out, like it's very difficult. So these are the cells which later can be eluted, and from there we do the mammosphere formation assay, like the 3D culture, then clonogenic assay, like where we grow them in soft agar and wait for 1 month so that they can form a clump. Then we study the migration assay, invasion assay. These are the few again data I'm sharing with you, where we can see when there is no platelets, like no treatment for luminal A patient and from TNBC. This is the mammosphere. And when we interact them with resting platelets, like giving aspirin, the mammosphere is not too big. But when we are interacting them with it's done from the same patient, like same patient blood, we have taken the platelet, and from them we have taken the stem cell, and we have co-cultured. So there you can see it's very big. So tumor educated platelets helps the cancer stem cell growth. This is the migration assay. These are the empty holes you can see, like in this case in Boyden chamber, we keep the cells on top of the membrane, and down we have treated the cells with platelets with aspirin, here without aspirin, and here you can see the more cells are have crossed the barrier. This is like normal, no treatment group. This is like matrigel invasion assay. So it's a marker of angiogenesis, where we have the matrigel plaque. Over that we lay the cells. This is with interaction with platelets, and this is with platelet rich plasma. This is without aspirin, and here it's like very angiogenesis is very high. And we have also checked it in vivo, where we have given these activated platelets plus tumor to the mice. That cancer progression is much higher in case of PEP, and there we have checked in EAC, Ehrlich carcinoma, and like Swiss mice and 41 breast cancer model. And we have checked the lung. We can see the foci, tumor foci, it's secondary tumor in lung. So these platelets, activated platelets, can are responsible in tumor metastasis. From there, we can see that. So again, like activated platelets are much higher amount in TNBC within the tumor, and they interact with CSC, cancer stem cell. And currently, we are studying that what are the exact molecular pathway involved in this interaction, so that we can target that. So maybe next time, if I get the opportunity, I can discuss. So this is my institute, Chitananda National Cancer Institute, Hajra campus, where I do all my work. This is the sixth floor laboratory from where I am doing. I am acknowledging Director CNCA for giving the facility, animal facility, member of surgical oncology, and of course the donors, patients with their permission, we got their blood and tumor samples. Without them, this work was not possible. I am also thankful to the funding agencies like ICMR, UDC. This is my lab. Today I presented the first part of the work from Jasmine Sultana. She is working with stem cell T cell interaction. And this is Aishwarya Gua, who is doing with platelet stem cell interaction. Thank you. I'm happy to take any question.
Thank you, sir, for the deep insights on the cancer biology and the pros and cons treatments, as well as the new dimensions of researches what you have shown from your research data, where the controlling regulations and the management of the cancer cells in your research institutes, how you are progressing towards. So I'm really sure that there are a couple of questions, and already questions are popping up in the window. So with your permission, and with the
Yes,
permission of
certainly, no
coordinator
problem.
and in the panel, we can take the questions for this particular interesting session. The first question which has been asked, it is by A. Madhavrani, Dr. A. Madhavrani. This 2D glucose, this 2D glucose is tumor specific, is it not? So blocking the glycolysis is it not blocking the glycolysis of the normal cell? That is the question from A. Madhavrani, the participant.
Yes, yes. Thank you very much for the question. Actually, of course, it is blocking, but in normal condition, we know we cannot deprive any cell from glucose. If it is living, it has to give in glucose. But if selectively, like if I have, instead of giving 100 molecule of glucose to cancer cell and normal cell, if I give 50 molecule of glucose and 50 molecule of 2D to a normal 100 cell, and a 100 cancer cell with 50 molecule of glucose and 50 molecule of 2DG, there you can see in normal cells, when they will not get, they will get 38 ATP from one glucose, and they will do their necessary proliferation. When it is not required, they will not divide, so they will not get stress. But in cancer condition, their main target is to divide. So they will take up the 2DG or glucose, whatever available. The glucose receptors like GLUT1, GLUT4 are very high in cancer. So they will just take up, normal cell will stop taking 2DG or glucose if it is not favorable. But they will take up more, they will take up more, they will having the trouble. In that way, they will be targeted. So we cannot deprive our body with glucose. So glucose has to be given, but if we can control it, like if we give, so that in that way, like 2DG is not toxic. And one version of 2DG is already in human use. Maybe few of you will know. That is, we do the PET scan. So there fluorodeoxyglucose is used, FDG. So that is having the radioactivity. Instead of oxygen, fluorine is there. So that is already is in use. So where again the same technique is used, where patient was given this injection. Before that, overnight he has to do the fasting, like 10 o'clock. They finish their dinner, morning don't take anything. So body is in high need of glucose, and then they inject them fluorodeoxyglucose. So all cells take up this, and the patient used to be sit for half an hour. So in that time, normal cell will remove it, but cancer cell will not able to remove it. The more they will take, they will keep accumulating this, and we can image it. So that is the mechanism for PET CT. So in that way, 2DG is not very toxic to human, and it's already tested even in during COVID. Also, the 2DG was used in some cases. So of course, it will block the glycolysis, but it will not affect normal cells that much than the cancer cell, because normal cell will not divide. So then they will have no problem. And anyhow, they are having some glucose to do their function, where they are getting 38 ATP, whereas in cancer cell it is getting only 2. So that's the thing. Thank
Thank
you
you,
for the
sir.
question.
Thank you, sir. And I guess Madam has already got her answer. The next question from Dr. Ashish Kumar Singh. With your permission, I would like to ask,
Yes, yes.
can we modulate immune response from pro-inflammatory to anti-inflammatory by selecting specific peptide which can activate MHC2 CD4 plus cell, producing the Th2 response? Your
Yes,
view
yes.
regarding the modulation?
Yes, yes, that can be certainly done, and many peptide therapy is coming also. Like we are also plan to work in that direction. So now the softwares are there. There you can like target. You can make your peptide like uh, 10 molecule, like 10 amino acid, 8 amino acid, 9 amino acid, and you can check their compatibility with the MHC, and then you can try it after doing this. Dry lab work, you can try it. And of course, peptide therapy is a upcoming therapy, and if we that can certainly be used.
Sir, another
You can
question from
immunomodulate.
Dr.
Yes, yes.
Dr. Abhijit Joshi. He is asking, sir, if we reduce the EMP or glucose uptake rate, it will suppress the cancerous cell activity or it will generate the ROS, reactive oxygen species.
Uh, so like reactive oxygen species like 2DG, that if continuously that is taken up by the cell, the ROS will be increased within the cancer cell, and that will give the ER stress, and cancer cell will die. And EMT, like it is very difficult to control, because there is no one mechanism is there that we can control that. Like many mechanisms are there, like it's a multiple problem from different sides. So if we can control the EMT, so like if we can block the angiogenesis itself, then many problem can be solved. But when even people thought of also like that, that if we can block the angiogenesis, we can directly start the tumor and kill it. But the main problem is that angiogenesis is a natural process. So like in patient, I told you, even like cancer surgery, that is one important thing. So to recover from that, angiogenesis is required. So if we have gone for surgery for a long time, we cannot give any angiogenic drug. Or if we are giving anti-angiogenic drug, we can't go for surgery because wound will not heal. So in that way, it's very difficult. So that's why it's not 100%. Like if we get this, we can block it. It's like that. So this is the like judicially we have to apply. So there is a concept of drug holiday. So someday it's not good to remove all sensitive cells. We give drug, we keep it under control, we leave it again, so that the slow growing, fast growing drugs, cancer cells, they will proliferate. So we can again give some drug, bring them under control. So if we completely remove them, then only the residual mass is there, which is resistant. So that's why after few cycles, we will see that the drug resistance problem comes, and then the normal cells are getting more affected than the cancer cells. So that's why like lot of modulation is taking place, and we are all trying to find some solution where we can balance it. It's all the matter of balance.
Thank you, sir. Another question from Nisargini Chotai, madam. Sir, does the platelets associated with tumor cells have any specific signaling mechanism or it could be random?
No, they have many specific mechanisms. So like I cannot tell you, as I told, it's under publication, I submitted it. So we are now actually trying out that what are the underlying mechanisms which are responsible And if we can find out those, like I can say till now, like beta-catenin pathway, Wnt beta-catenin pathway, that is one major pathway which are active in platelet cancer stem cell interaction. So if we can block that pathway using some small molecules, then yeah, like one way we can block it. Other way, we have to stop the platelet itself from being activated so that it cannot do its the function what it is doing. And in that case, aspirin is coming up as a very good solution because it is again very cheap and already tested in humans. So it is all repurposing. Many drugs are nowadays we are repurposing for this, like metformin is repurposed, then like statins are repurposed for cancer because they are already, so there are multiple targets. So in that way, if we can see the patient properly, if you can know their proper health condition, then we can use this thing to modulate their immune cells, the best way to modulate their immune cells so that that itself can kill the cancer, has the potential to kill the cancer.
Thank you, sir. Lakshmi Yadav, one participant is asking, sir, what are the parameters that are used to detect the stage of the breast cancer?
There are many things are there, like best thing we have to do the histology. So that is the final thing, because even you do USG, X-ray, MRI, PET CT, they will be telling that this may be a cancer and it has to be correlated to the cytology. Like FNAC or liquid biopsy is the main thing which will tell in which stage it is, what is the its ethology, like from which tissue is that it arises and how much it is differentiated. So if you see any result, like TNM classification is there. So tumor size, then its origin and metastasis, how much it is differentiated. Is it poorly differentiated? Means how much it looks from the different from the normal tissue. It's moderately differentiated. It's highly differentiated. So in that way, like the histology is the main thing which will give, and TNM, that is the size of the tumor, metastasis, how many nodes are involved. Like if it is gone to any lymph node nearby, so if it has not, so it will be one layer. If it has gone to one lymph node, then it's progressed. If more than one lymph nodes are involved, then it's more advanced. And if it has gone out of the organ, like any other part, then it is like from breast cancer, it goes to medial sternum and all. So it goes to bone nearby areas. So then it is like if metastasis like M0 or M1, even small thing is gone means we don't know exactly where it can go, because whole body anywhere it will go. Though there are some seed and soil theories there, like we know that where it can go. And the most of the secondary metastasis you will find either in lung or in bone, because these are the two very organ which is very highly oxygenated even and having lot of nutrient, and they also have the structure like a scaffold. So if you see the bones structure there, you can stick. There is a position where it can either it has to lodge itself, and then you can get the nutrient. And the same way, lung also you will see lot of gaps are there, alveolar cell. So they stuck there and starts making their uh, secondary sites where you will find very less cancer. Like it's very rare in heart. Very rare it happens because like exactly we don't know why it's not there. But these are the part we know that it goes to spleen because again it's drainage are there, different lymph nodes. So it all depends.
Hello, ma'am. Are you there, madam? Okay. Okay. So, just a second. Yeah, I think ma'am has just now disconnected because of some kind of internet issues. So once again, I thank you so much, Dr. Banerjee, for taking time from your busy schedule Okay, since I am actually personally connected to many of you, okay, I know your individual responsibilities and the task. So I just want to let all the participants know here again, like some of our other resource persons, Dr. Banerjee is now very involved in some kind of conferences, which their institute is hosting, and it's
Yeah, we
quite
are hosting
important.
ICR 2025 in Calcutta, and that is from 16th and 15 is the pre-conference workshop. Again, I am organizing the flow cytometry there. So till morning we are preparing the guest list. I am in the travel accommodation. So who will stay where, and we are making our in between. So anyhow, this is also part of our work, I think, because whatever we know, if we don't share, then we have to also encourage others to, so that they also can work together, and there is maybe collaboration work can be done anywhere. So we are open to all, and I'm very happy to give my presentation here. Hope you all liked it.
Yeah, I think it was really very interesting, and we can see, by the we can judge by the questions, the responses of the audience here. So once again, we are really grateful to you, okay, for this very nice presentation and
Thank
research.
you for
So
giving me the opportunity to present a few of my work and discuss the thing for such a huge number of people all over India. I'll be very happy to
yes, it's
take
always
it
our
anywhere.
pleasure, we
Okay.
keep looking for this kind of collaborations and connections. So
Yes, yes.
hope we can again connect soon. Okay, like this in the future.
Okay.
And
Thank
thank you
you.
so much
I
once
will be
again.
always happy to present, like it's my pleasure. Thank you.
Yeah, yeah. So once again, on the behalf of Atmia University, this Atul LTP program, and the coordinators, myself and Moshri Madam, we thank you, okay, for coming here and delivering this very, nice and thought-provoking topic. So, पार्थ भाई एक अच्छा सा राउंड अप्रोच हो जाए सर के लिए. So let us all also join.
Thank you. Thank you.
Okay, fine. So, Dr. Shabdak, you can you can be joined here, or as you're telling that you are very busy at the moment. If you
Yes,
want,
I have to
you can
again
take
start.
me.
Like till
Yeah.
like I am attending most of your sessions. Like yesterday also I was there. Day before yesterday also there. I was listening to Ratnadeep. It was excellent presentation and all. And later I listen the recording part, so that because it's very different type of work, so you should know that what they are doing all over the different people. So it's excellent, like thing for. I'll be very
Thank
happy to be a part of this, and I will keep listening. Don't worry.
thank you again for your kind words. We have tried to incorporate from various kind of diverse kind of research areas and
Yeah.
topics so that it can give overall view to all our participants.
Yes.
Okay, so with this words, we'll move on to our next speaker for today. So we are again, I am privileged to welcome the next eminent resource person for this FTP today. His name is Dr. Hiram Kulkarni, and he is going to present on the topic entitled HRMS and the critical quality attributes of protein of interest. Before he actually starts his talk, I just wanted to give a brief overview of the credentials of Dr. Kulkarni. So, Dr. Hiram Madhav Kulkarni has earned his PhD in life sciences from Center for Cellular and Molecular Biology, CCMB, Hyderabad. He has over 9 years of industrial experience now after his PhD. He has been working in different kinds of organizations actually, and very recently he has joined as scientific officer at the, again, cancer research institute called ACTREC in Navi Mumbai. So we are we are really grateful that both the speakers are will talking, are related, associated with Cancer Institute. Dr. Kulkarni's main areas of interest again are preclinical and analytical R&D, biopharmaceuticals, proteomics, etc. So now I welcome Dr. Kulkarni to kindly present his slides. पार्थ भाई आप डॉक्टर कुलकर्णी प्रेजेंटर राइट्स दे दीजिए.
Hello,
Yes,
am
yes,
I
Dr.
audible?
Heron. Yes, you are very much. Just see that if you can share the slides.
Uh, yes, I'm trying that. I've been given presenter rights, right?
Yes, I I I think there is some kind of issue. Maybe it's gone now. Uh, just hang on for a moment. I will look
Yeah,
into it.
yes,
पार्थ
yes,
भाई,
yes.
पार्थ भाई आप सर को दे दीजिए प्रेजेंटर राइट्स. आई थिंक अभी उनके पास नहीं है प्रेजेंटर राइट्स.
नहीं सर, प्रेजेंट राइट दे दिए है सर जी.
ओके.
Okay, I'll try just once again.
Yes, Dr. Hedder, you have the presenter rights actually. Yes. Now,
Okay, now,
um,
okay. Now my screen is visible, right?
yes, your screen is visible, but yeah,
Yeah, I'll just open this thing.
yes, no problem.
So, I'm using a PDF version of my presentation because there is some problem with the PowerPoint that I have prepared.
Okay. Do you want us in any way to present your PPTs from our end, or you're fine and comfortable with this?
I think whatever I'm sharing right now, the title slide, this one is visible, right? There is no problem with this.
No. It is visible. Fine. You can carry on.
Okay, yes, great. So, good evening, everyone. Thanks, Dr. Devashish, for introducing me with kind words. So, the title of this talk is HRMS and the critical quality attributes of protein of interest. Now, HRMS is high resolution mass spectrometry, and I have chosen to call this particular thing as protein of interest because uh, I would mainly focus on a protein which could be used as a biopharmaceutical product, but the same things could be extended to the proteins which are used for other industrial applications, like for diagnostic purpose or veterinary purpose, and so on and so forth. So, since I have been working so far in in industrial research environment, I do not have data of my own, like all that is guarded with confidentiality statements and everything. Whatever I would be presenting is from public domain, and I have made attributions wherever possible. And from those attributions, you can always find the original figures or informations or everything that is necessary for further those who want to carry that further. So I'll start with quality by design. Okay. So the title of the talk is HRMS and critical quality attributes. Now first I will come to critical quality attributes. So quality by design is a concept in management. Dr. J.M. Juran is the pioneer of it. He's no more with us. He's one of those 20th century stories in which some Jew somewhere from Europe migrates to US and becomes one of the torchbearers of the field. Okay, so Dr. Juran is one of such people who rose from some persecuted group of people, and he achieved what has become a beacon for everyone now. So this particular quality by design concept includes three things. This is called a Juran trilogy. First thing is quality in planning. So if you want a quality product or quality service at the end, it starts from the planning. So you have to plan quality everywhere. Like you people also might have heard total quality management and similar terms. So quality in planning is very important. You have to define your targets very clearly, and you should have a structured plan of achieving those targets. Second thing is quality control in processes. So you have to keep checking the processes so that they work reliably. You should have a quality control and a dedicated quality control team which takes care of that. The third thing is quality improvement. So you have to keep learning from your failures. You have to keep learning from your experiences and keep refining the quality. So uh, like there is no end point to this process. So in 1960s and 70s there was a quality revolution in Japan. Many of you must be aware of this thing. So before this revolution, Japanese goods was not considered to be of a very great quality. But after that particular revolution, which included this third thing, quality improvement, continuous learning and continuous refining your product, has brought Japan to such a level that like it is it has become granted that if the thing is from Japan, it must be of the highest quality. So now this quality by design concept has been by default applied worldwide for pharmaceuticals, because pharmaceuticals are directly concerned with the life of the patients. So it is a life and death situation, and we cannot have uh, risk for that particular, like the quality of the product. It always has to be of the highest quality. So especially US FDA by mandate has accepted quality by design approach, and that's how worldwide it for pharmaceuticals it has become a thing to follow by mandate. It should go through quality by design approach. What is quality? Quality is fitness for purpose. So please remember this: quality is fitness for purpose. And uh, here most of the times I'll be referencing to the US FDA guidelines. Believe me, other guidelines from other regulatory bodies like Indian FDA or Brazil or European EMA, all these regulatory bodies more or less follow the similar pattern. So, like the particular examples I'm giving are from US FDA's guidelines, but they would be in general applicable to everywhere. So, this particular, you know, pharmaceutical development starts with defining the quality target product profile QTPP as it relates to the quality. That is, in case of a pharmaceutical product, which here in the scope of our talk, it will be a protein, it will relate to the safety and the efficacy of the product. So these two things are very important for a pharmaceutical product, which is a protein, has to be safe and it has to be effective. Safety and efficacy are two important things over which its quality depends. So, identifying potential critical quality attributes of the product depends on this QTPP, this target profile. You have to identify the quality target product profile, and from them you have to design your critical quality attributes. That these are the attributes that I would be checking for the quality of the product. Okay, so these characteristics uh, have an impact on the product quality. That means on safety and efficacy. You have to identify and enlist those products, and you should have a standard and design planned way of testing those critical quality attributes. So this is a in nutshell, this is how the quality of a pharmaceutical product, here in the scope of this particular talk it would be a protein, is controlled. Okay, so some of these CQAs, critical quality attributes, are to be assessed for every batch release, and some are to be studied during the development phases. And when you perform a regulatory submission, you have to provide data for them. And both of them are called CQAs. They should be like, for both of them they should be there should be a validated process or a validated method which measures that particular attribute. So here is this whole thing is carried out under quality risk assessment. So here is an example from US FDA's quality by design for abbreviated new drug applications uh, guidance, where you have to properly prepare a matrix. Risk assessment should be well documented with the assumption and rationale behind every ranking. So you have to rank them. For example, in this particular table, it has been ranked as low, medium, and high. Okay. So these assumptions and rationales should be there in written form. They should be well documented so that even like it shouldn't be just empirical. And the assumptions and rationales can be from the available knowledge, from the experience of the personnel who are involved in that, or from the fresh experimental data generated in your lab. And periodic review of this ranking is desirable because the field is constantly changing and evolving. And there is always new research, there is always new research paper on the net, and there is always a new technology which can measure it to a better refinement. So a periodic review, quarterly or 6 monthly or something like that, is always good, so that the criticality can be adjusted to the current state of knowledge, to the very latest state of knowledge. So coming to the critical quality attributes, all these things has been covered under the Public Health Act of United States. So anyone who is interested of about the legal mandate there can get a copy of that from the internet anytime. So the critical quality attribute has been defined in ICH guideline, ICH Q8R2. CQA is a physical, chemical, biological, or microbiological property or a characteristic that should be within an appropriate limit, range, or distribution to ensure the desired product quality. Then CQAs are generally associated with the drug substance, excipient, intermediates, in process materials, and drug products. So this particular approach can be used at any stage of development. May it be just developmental, it should be at the pilot level, or it should like I already said, like just before, like if you're bringing that thing to market, just before every batch release also you'll have some list of critical quality attributes that are to be checked. And release testing requirements have been described in USFDA's industry guidance Q6B. Those are helpful. They discuss about safety, purity, identity, and potency of the product, and they have a structured way of addressing these things, or establishing rather these things. And US Code of Federal Regulations 21 CFR 610. This is a subsection of the same act that I was talking about, the Public Health Service Act. So this particular section 21 CFR 610 describes it covers this part. And the selected attribute should be measurable and not empirical. This is very important. For example, if you select certain attribute, okay, uh, that for example, the product shouldn't smell or something like that, which depends on the senses of the person who is involved there, okay, or the product is clear to the vision, something like that. Okay, that just like on many certificates of analysis we do have this kind of descriptions that clarity, yes, or transparent, yes, something like that. So these things depend upon the sense of the person who is actually performing that particular test. So this becomes empirical. So a critical quality attribute should not be empirical by convention, it should be measurable. Of course there will be exceptions here and there, but an attempt should be made that it has to be measurable, and there should be some acceptance range associated with it, which is predefined. And it has that testing should be validated. Okay, the method should be validated. So ICH guideline Q2R2 provides a good description about uh, validation of methods that can be used in analytical testing. The CQAs and their limits should be science driven. They should be customized for each protein product, and each product has their own list of CQAs, though many of them may be same. For example, like it is enticing that your lab is already working on some protein, and you just use the same critical quality attributes list to some other protein that you are trying to develop. So it lessens your work, but there are pitfalls there. You have to study the new protein from the beginning, and you have to form its own CQA list. For example, I have come across a certain protein. Some lab was trying to develop a protein, uh, and in that particular sequence there was no cysteine in the entire sequence, amino acid sequence of that particular protein. And they were trying to find out a disulfide map of that particular protein. So now those who are you know uh, who are privy to protein science know that a disulfide bond requires at least two cysteines to be present there. So in that particular thing shouldn't have been a CQA there. But this kind of things happen, and if they are not corrected on time, it leads to a lot of confusion, a lot of waste of resources and time and money, of course. So it is always good that CQAs and their limits should be designed separate for each product, like radically separate, approach has to be taken for every product. So they say that a stitch on time saves nine. So it is this kind of thing. So what practically happens is, uh, since, like, especially if you're not working on a biosimilar, and you're working on some novel protein, in the beginning you don't have enough information to design the CQAs. So you have to start with potential CQAs. So you have to find the available state of knowledge, available technology, then data from your own experiments, data from experience with other molecules, and regulatory guidelines. You have to study all of them, and you have to prepare your own list of potential CQAs. Some of them, it's a pool, and like it keeps changing uh, when you go on developing that molecule and gather more and more knowledge about that particular molecule. So here in this particular talk, I'll be concentrating on high resolution mass spectrometry and the CQAs which can be addressed, the general CQAs which can be addressed by using high resolution methods. So the CQAs, those could be addressed by high resolution mass spectrometry. I have selected those which are kind of applicable to most of the proteins. But as I said, uh, whenever you have new product, you have to always study it from the beginning and check how that particular CQA, like whether it has to be there in the first place, and second thing, how it can be addressed. So here are some of those potential CQAs: primary structure, uh, you know higher order structure, host cell protein, post translational modifications, and the titer. Okay, so primary structure involves intact mass analysis by HRMS, peptide mapping, post-translational modifications analysis, charge variant analysis, etc. Uh, Higher order structure, this subunit, if there are more than one subunits in your protein, for example, a monoclonal antibody of IgG, one class will have two small chains and two big chains, heavy and light chains. Then hydrogen deuterium exchange can help you find out more about the secondary and tertiary structure. Disulfide mapping provides a framework for its folding and all. And host cell protein analysis shows whether there is something more in your preparation, like some contaminants or something else, uh, because many of these proteins are prepared by biotechnological methods. Most of them, actually, I wouldn't say many, except some molecules classified as peptides, which are chemically synthesized. Other proteins are synthesized by biological means, and there ought to have some lingering proteins from the system in which they have been expressed. And last but not least is quantization. So it can be done in targeted and untargeted mode using high resolution mass spectrometry. Quantization is important because for the efficacy of the product, the that particular preparation has to be of a certain concentration. It has to have a certain titer of that molecule, and it has to be tightly controlled. Okay, so again, here's the scope of the this particular session. So I'll be talking about, like the thing is that HRMS, high resolution mass spectrometry, is a technology, and the regulatory and management requirements are safety, purity, identity, and potency. Okay, so we have to find out what methods in the available technology address these issues. Okay, so there is like each of them may be covering one or more of these particular things in their own particular fashion. So it is difficult to find one to one relation here. Okay, so I'll be discussing about certain techniques which are used in resolution mass spectrometry, which include intact mass analysis, subunit mass variants, targeted quantization, PTM, disulfide mapping, host cell proteins, peptide mapping. Okay, out of all of these, intact mass analysis and peptide mapping are the ones which I would be describing in good detail, and others, once you understand these two things, other things are easy to understand. Like they are just customized applications of the same things. In this particular talk, I won't be talking about hydrogen deuterium exchange and cross-linking mass spectrometry, which are used for secondary and tertiary structure analysis of the proteins for two reasons. First, they so far I haven't seen them being used for regulatory submissions. Okay, there are other orthogonal techniques which are like currently more reliable than these two. Uh, of course, if anyone of you is interested, I would be happy to share research articles or method articles with them. But I have just skipped them from here because I don't think they would be as useful as the other. Okay, so coming to the structure of proteins, uh, those who are acquainted with protein science, like this is a the thing from uh, Leninger's book, biochemistry book, which I think everyone has gone through. So it consists of primary structure, which is amino acid residues, like a chain of amino acids. Then it folds into a secondary structure, which is like alpha helix or beta sheet, or there are different types of beta sheets and turns and all those things. A tertiary structure, and if there are more than one subunits, there will be a quaternary structure as well. So high resolution mass spectrometry helps us uh, on all these levels: peptide mapping and de novo sequencing. These things provide us the primary structure of the protein. Hydrogen deuterium exchange and cross-linking MS can be applied to all the higher structures. Intact mass analysis can provide you the exact mass of your protein in intact form. Subunit mass analysis provides you masses of the subunits of your complex protein. Disulfide mapping provides you the exact map of which cysteine is bound with which cysteine, and where the disulfide bond is actually being formed, so that you can have a proper frame of the folding of your protein. So let us start with intact mass analysis. Intact mass of a protein is the mass of the protein with all its covalent bonds intact. So this includes the post-translational modifications, the disulfide bridges, and everything. If the sample preparation procedure does not affect the mass, then we can afford to let go the secondary and tertiary features in sample preparation, because we are only interested in the intactness of the molecule on covalent bonds level. So this particular thing provides identity of the protein. It also provides us presence of isoform, proteoforms or splice variants of your protein. If they are present, they can be easily detected at this stage, and impurities if they are there. So intact mass analysis here is I'm just yeah. So I'll be showing you example using electrons, ESI source, electrospray ionization source. So this particular thing can also be done using Maldi source. I'm aware of that, but here I'm just describing the most prevalent uh, technique that is being used in industry. So in electrospray ionization, the glass capillary sprays a sample, which there is a Coulombic explosion and all those things, expansion and all those things are there. Your molecule basically gets charged, it acquires some protons, and it is sent into mass spectrometer to mass analyzer, where what you find is mass by charge ratio. Okay, so you, for a single protein, you get a complete look at the panel B, you get a complete distribution of different charge states of that particular molecule. A mass spectrum is relative intensity. It can be absolute intensity versus mass by charge ratio. So for a given protein, you will find multiple charge states. Here is how an ESI spectrum actually looks like and how to read it. Okay, so y-axis shows you the intensity. It could be relative or it could be absolute. X-axis shows you mass to charge ratio of that particular molecule. The peak, which is of highest intensity in your spectrum, is called as base peak, and the intensities of other peaks are relatively counted from like calling the base peak as 100%. Absolute count of the base peak you should find somewhere at the top of the spectrum, or this thing actually is fluid. It depends upon which vendor and which software you're using to see your data, but somewhere on the in the spectrum, at some corner, you should find this particular thing. What is the absolute count for your base peak? And then there will always be other peaks which you would be interested in. And the charge state, like we would be interested in mass of the molecule, not mass by charge of the molecule. So we need to find m. For finding the m, we should be knowing z, because what we have is m by z. If we find z, we can find m. M is molecular, like mass of that particular thing, particular entity. So here is how we can find the charge state of your molecule. This is a distinct advantage of high resolution mass spectrometry. In case of low resolution mass spectrometry, you won't be able to find this. So in high resolution mass spectrometry, you get a resolution to a level that the isotopic peaks of your protein will be separate, or like your molecule will be appearing separately. They would be resolved properly. If the charge is 1, the difference between them has to be at least 1 Dalton. Okay, and if the difference between these particular isotopic peaks is less than 1 Dalton, that means there are number of charges there, more than one charge there, because an isotope cannot appear without addition of at least one neutron. So, for example, if there is a difference of zero point 0.5 Dalton, that means that particular peak is showing charge state of 2. If the difference would have been 0.33 Dalton, it would have been 3, and so on. Now, the mass by charge to mass calculation can be performed uh, by this particular simple formula. Mass by Z is equal to mass of the molecule, capital M here is the mass of the molecule, plus Z into X, the mass of the adduct ion. Now, as I told you in the beginning, that you are adding some protons. I'm talking about a positive, charge mass spectrometry here. Uh, so since I am talking in terms the scope is limited to proteins, I am directly taking it granted that we will be adding protons here. A negative mass spectrometry can also be done, but that is a topic of a different talk altogether. So when we are adding adduct ion, it could be protons, it could be ammonium ions, it could be some other metal ions. Whatever adduct ions we are adding onto that particular molecule, they also have their own mass, molecular mass, and that also comes, that gets added to that particular ion, and it shows up in your spectrum. So you have to add that in your mass of the molecule, m plus zx divided by z. So z is the number of charges. So if you want to solve this particular thing for capital M, which is mass of the molecule, you have to rearrange it in this way. You, from the spectrum, you get mass by charge ratio. You have to multiply it with the charge that you have identified, and you have to subtract the mass of the adduct ions. Now, after that you get the mass of the molecule. Now, error of measurement of charges with each additional charge uh, is different, and here is the catch. So what happens in case of proteins is, uh, as I showed you earlier, there will be a complete distribution of different charge states, and you will have so many different peaks, and the error associated with each of them will be different because it is different with the changes with additional charges. So it becomes very difficult to establish a particular single mass from that very complex spectrum. You can do it easily. You can solve this particular you know equation in your mind for a small molecule, but for a protein it would be a very difficult task, almost undoable. So that's why here a particular process is used, which is called as deconvolution. So here, uh, small f is the distribution function for the peak heights in the measured spectrum. So a particular, uh, like each of the charge states will provide you a particular result, and all those will be added up in a weighed sum of uh, uh, those charge states. So higher is the height of that particular charge state, more it will contribute to the final result. So this thing is done using softwares. A lot of computational help is available for this. The original like I would recommend all of you to go to this particular reference, Matthias Mann. This has been first done in 1989, and they describe it very beautifully. And from then on, deconvolution has become a standard part of all the software packages from all the vendors. So for having, for deconvoluting your spectrum, you need three things. The output range in Dalton. You should have certain idea of the expected mass of your protein. For example, if you are working with some protein of say 50 kilodalton, and if you put a range of something like 10 to 40, you are not going to find your answer. Or even if you put a a range 10 to 100,000 daltons, you would assume that my 50 kilodalton protein is anyways covered here. But still, since your range is very big, there could be problems in finding the correct mass there by deconvolution. So knowing the output range is very useful here. The probable adduct ion, because this adduct ion mass will be deducted at the end. So generally people try to keep these product ions protons, which becomes very easy, but sometimes it is not possible. So you must be knowing what probable adduct ion is there, It could be ammonium, or it could be sodium, cesium, or no, any positive ion could be there. Then the possible charge states, this thing also you should be knowing. So generally, if you know the mass of your expected protein, like the output range, then you can also kind of judge what are the possible charge states from your spectrum. If all these three things are known, you can properly perform the operation of deconvolution, and you can find out the right answer from this. So I'll come to subunit mass analysis, which is kind of uh, customization of the same workflow that I have discussed so far. So this is nothing but intact mass of individual subunits of a protein. Okay. So I am just showing you an example of IgG1 molecule, which consists of two light chains and two heavy chains, and they are bound together by specific disulfide bonds. So if if you want to completely characterize this molecule, it is not enough that you just have the intact mass of your molecule. You should also be knowing the masses of both light chains and both heavy chains, because it decreases the quality risk there. Like, it is always possible that the intact mass matches very well, but that was just because the errors in masses of heavy and light chain actually cancelled each other, kind of thing. So they, I'm not sure this thing, but about this thing, but they call it uh, error type 3. Like, you have a right answer, but actually it is wrong. Okay. So it provides you more clarity on the tertiary structure. It so in case of biological molecules, it is very important for proteins which are having subunits. You should always go for uh, subunit mass analysis. Another famous example for this is insulin. Insulin has two chains which are bound with disulfide links, and if you're working on insulin, it is mandatory that you should show this thing, uh, the subunit mass analysis. Now, separation of subunits can be done by breaking the disulfide bridges. You can reduce them or you can oxidize them. So mostly people go by the reduction way. They reduce it by using certain reducing agent like uh, dithiotritol, DTT. This is what they call it. And then they are further derivatized. Like, there will be two SH groups, thiol groups, which are reactive and which can form the bonds again. And this time they will be scrambled. So uh, these thiol groups are further, derivatized by using iodoacetamide, so that they do not form the bond again. Now, while working on the spectrum, you have to along with the adduct ions, you will also have to consider the mass of the added group here. And there are now more reagents there, like the push is towards greener chemistry. So TSAP and uh, there are many more reagents that are coming up which can replace dithiotritol. In fact, earlier mercaptoethanol used to be used, beta mercaptoethanol, but now it is almost out of use. Now here, since you will have more than one entities in your product, once you separate the subunits, it is always good to separate them, because what happens is there will be a phenomenon of charge competition in your electrospray ionization source. So if more than one things enter at a time in your source, they will compete with each other for the charge, for the protons or whatever adduct ions you are trying to put onto them. And one of them or some of them might get suppressed, and some of like others will be over represented. So to avoid that, it is good to separate the subunits before feeding them to mass spec. So separation of interfering substances from the molecule of interest is needed. Like for each subunit, the other subunit is a interfering substance here. So reverse phase, liquid chromatography is a very reliable technique. It has been proven, and for protein separation, and it is very easy to develop. It is a robust technique. So reverse phase separation of subunits is recommended here. You can in fact use reverse phase in your intact mass analysis where there is no separation of subunits. Even there you can use it, so that if there are proteoforms or other things available in your product, they also get separated over the LC, and you get complete information. So here I'm showing just an example. So if you have a mix of certain proteins, for example, insulin, glucagon, myoglobin, and BSA and cytochrome C, okay, if you try to inject them at a time, you won't be finding anything. Okay, like hardly you will, like if you're lucky, you will find one of those molecules, otherwise you won't find anything. But if you separate them on LC, they appear at different RTs, enter the mass spec separately, and then you can beautifully find out their masses. So this example is from an application note from Agilent. So the general workflow for intact and subunit mass analysis for adalimumab, like it can be used for any protein. Here the example is of adalimumab. So get a pure product, then reduce it. Reduce if you want to perform a subunit mass analysis or skip this particular step. Then go to chromatographic separation and get your spectrum. You will get intensity by M by Z, and then from there you perform deconvolution and other data operations, and you find the mass or the masses of the subunits. Okay, now let us come to peptide mapping. So the amino acid sequence of the protein is important for its identity and biological activity, and so efficacy of the product depends on it. Actually, the identity of the protein is the amino acid sequence of that particular protein. So definitely, if the sequence is not right, we won't call it the protein. Like we won't call it a right protein. The incorrect sequence, if there are deletions, if there are replacements or other kind of things, it may lead to aggregations. It may lead to elicit unexpected immune responses, and that's why it is a big safety risk also. So the amino acid sequence of your protein or the primary structure of your protein has to be flawless. So that's why actually US FDA's guidance mandates that you should show the amino acid sequence of your protein to the extent possible. Now they do not actually mention a certain number here, but the available technology gives you 100% sequence coverage. So basically it means that whenever you are trying to develop a new protein product, you have to show 100% sequence coverage, and from first amino acid to the last amino acid, you should be able to show that the amino acid of your sequence, your protein, is exactly the one which you are expecting. So the PTMs also contribute to the efficacy of the product. For example, some of the glycan molecules are, they define, the their access to certain hydrophilic and hydrophobic areas, or their half life in the system of the host. So the PTMs also definitely contribute to the efficacy of the product. Sometimes they can also be a safety risk if they are not expected there. They can elicit immune response. A wrong, a very alien kind of glycan can actually elicit immune response. And the procedure, the peptide mapping procedure uh, involves digesting of the protein in smaller peptides, and then we subject them to mass spectrometry analysis. Now here I'll be introducing you to the tandem mass analysis, like MSMS analysis. So here is the general peptide mapping workflow. So this one is from a very old reference. So in those days, only trypsin was being used. Mostly trypsin was being used to digest the protein uh, at smaller peptides, and actually the protein sample would be taken, it would be denatured, it will be reduced and alkylated so that the disulfide bonds are reduced and alkylated, and then a buffer would be added to dilute the denaturant, and then trypsin would be added. Trypsin was like there were other chemical products as well, but trypsin was known to cut the protein at lysine and arginine, at the C terminal ends of lysine and arginine amino acids, very specific. Then overnight it will be kept for digestion. Quench it with formic acid. Trypsin actually works in basic environment. It requires pH of 7.5 to 8. So you quench it using formic acid and then analyze. This is how it was being done. Of course, over the time, several new enzymes have been discovered which are very specific. So sometimes what used to happen was trypsin was not enough for generating the peptides of optimal size, which can be detected and sequenced in mass spectrometer. So in mass spectrometry, by using uh, MS1, which is like similar to what we saw in intact mass analysis, it will be injected, like it will be like the peptides which are generated there will be separated over reverse phase chromatography, reverse phase column, and they will be fed to a mass spectrometer, and then uh, each of them will we will get a mass of each of them, and each of them will be further subjected to fragmentation, and now a spectrum of fragmentation of each of them will also be recorded. This second thing is called as MS2. Each of these peptides will be fragmented, and a fragment ion spectrum will be recorded, and from this fragment ion spectrum actually a sequence will be deduced for each of the peptides. If we find out sequence of each all the peptides which are generated, we effectively get the sequence of the entire protein. Now here one important thing you have to remember is, if you're working on a product which you're going to bring to market, you must be knowing its sequence beforehand. If it is just in R&D state, if you're just trying to find out a sequence, you can perform de novo sequencing, but mostly once you know the sequence, you will be using that particular sequence and trying to match it against your data by using specific softwares. This particular approach which I am showing you is called as bottom up proteomic approach. For those who are interested more deeply into it, this particular sequencing can be done by top down method also, which I'm not discussing here because it is not so far it is not very prevalent in this particular industry techniques. Now here tandem mass spectrometry actually paved the way to connect the sequence of the peptide to the mass spectrum. Peptide mass fingerprinting I will actually show you an entire figure and the history how it developed incrementally from several different discoveries in the field of mass spectrometry and protein science. Search algorithms based peptide mapping combined the digestion of the protein, tandem mass spectrometry, and the computational power. So this is how it developed. So first of all, some proteins used to get sequenced by the Edmond degradation, and you might have learned about those techniques. Soft ionization MS was found in 1989 or around that time, basically after 1985. Soft ionization technique was important in while you work with biomolecules, because the hard ionization techniques which were used before that prevented or the use of those techniques was not very successful when you were working with biomolecules. So the discovery of soft ionization techniques in mass spectrometry, like matrix associated laser desorption, MALDI, and ESI, electrospray ionization, were the groundbreaking things which allowed analyzing proteins or peptides using mass spectrometry. Sequence of proteins were found by Edmond degradation and some other methods. Then after that, genomes were being sequenced. So Saccharomyces cerevisiae or S aureus, Staphylococcus aureus were some of the earliest sequenced genomes. And once the genomes were sequenced, there was a new idea that these genes or gene sequences could be just translated to proteins, and we can use like we don't have to actually sequence the proteins. We can find the sequences of the proteins from the sequence of the genes. So the genome sequencing has been one important event here. Then site-specific digestion was discovered by trypsin and many other enzymes and chemical entities, which gave an important, a tool of digesting the proteins and, uh, bringing it to smaller peptides, which can be further sequenced. The understanding of PTMs went on growing, in the field. And the peptide mass fingerprint was first developed, in which only MS1 used to be performed. Like you would use the sequence of a protein, digest it with site specific protease in silico as well as experimentally. Now in silico you can predict what are the masses of the peptides that would be generated, and in your experiment you would be looking for those masses. If those two are matching, like the identity of the protein is confirmed. This is what is peptide mass fingerprinting. So the idea is that the mass spectrum of your digested protein is a fingerprint of your protein. You can identify your protein from it. Later on, tandem MS got added to it, where each of those peptides which are which appeared after digestion of a specific protease were further sequenced using fragmentation and like fragment ion spectrum. And of course the computational power went on growing uh, since last 30 years or so. Like of course everyone is aware the amount of computational power now available to us was not there in 90s. And so with the growth of computational power, all these things have become very automated. We can handle the data more confidently, and that's why peptide mapping and many other associated things have become very easy for us now. So here I'll just explain you what is tandem MS. So what happens is your sample, your molecule enters into the mass spec. It gets ionized at the source. Then it goes to mass by charge separation by the MS1. Like this is a mass analyzer. Whatever mass analyzer you are using, your molecule will be separated on the basis of mass by charge. Then it will enter a collision cell. Here it will be fragmented. Now I am just showing you the easiest one, that is collision induced dissociation. There are different types of fragmentation techniques. I'm not getting into the details of those right now. Just remember that your molecule will be fragmented by the use of energy or collision, and then this particular fragments will again be separated over m by z scale, and each of the each of the m by z separated here will have its own fragment ion spectrum again. So like this goes to detector, where the ms and msms spectra will be recorded. So the sequencing of proteins by tandem mass spectrometry was developed by you know uh, uh, John Yates, who is a big name in the field of biological mass spectrometry. So this particular paper is from 1986. So since then they are working on this particular thing. Here is one example. Uh, this left hand side panel shows a MS spectrum of a certain protein. Okay, so all these peaks you see in the left hand side panel are a different different peptides of that particular protein, and uh, one peak selected for MSMS. I am showing an MSMS spectrum of that particular peak on a right side panel. Now this right side fragment ion spectrum is generated by breaking this particular peak, which is of mass 1567 point something, and uh, you must be noticing certain notations here like y1 y b5 y4 and so on and so forth on the right hand side spectrum. So this is a way how the ions are named in fragment ion spectrum if the fragment ion spectrum is from a peptide. So finding a sequence from a tandem ion spectrum is like a jigsaw puzzle. You have a set of the fragments, and now from those fragments you have to build back your original molecule. Like you are going to find out what amino acid is at the n terminus, what is at the second position, so on and so forth till the last amino acid at the C terminus. Now if you check the backbone of a peptide, it consists of three bonds which keep on repeating. Okay, the first one is between the bond within an amino acid between the nitrogen and central carbon of an amino acid. The second bond is between the central carbon to the CO group carbon of that amino acid, and third one, which is most important one, is a peptide bond which is between the CO group, carboxy group of one amino acid residue, and the amino group NH of the next amino acid residue. A bond between these two is peptide bond. So whenever you fragment a peptide, uh, if it has to be fragmented, it would be a random fragmentation, and it has to get cleaved at one of these bonds. So since there are three types of repeating bonds, they have been named in a certain way. If the bond between the central carbon and the carboxy group carbon of an amino acid breaks, the N terminal portion of that particular peptide, a fragment forming from the N terminus is called as A ion, and the remaining portion, which is from the C terminus, is called as X ion. Similarly, if peptide bond is broken, the part towards the amino terminus is called as B ion, and the part towards the carboxy terminus is called as Y ion. If the bond between central carbon and the amino group nitrogen is broken, the N terminus part is called as C ion, and the carboxy terminus part is called as Z ion. So, in collision-induced dissociation technique, which is one of the oldest fragmentation techniques and very reliable, which people generally use very routinely, in this particular technique, the prevalence of B and Y type ions is very easily observed. So, mostly the peptide bond will be broken there, and you will find B and Y type ions. Now, since it is a random type of fragmentation, any of the peptide bonds can break. Okay, so you will find different a set of different fragments, and the difference between, like for example, if you are observing Y ions, just look at the left hand side panel. If you are observing the Y ions, okay, the difference between the successive Y ions will decide the identity of the amino acid at that particular place. For example, if you find if you start with precursor mass, like in tandem mass fragment ion spectrum, generally you will find unfragmented original mass to a certain level in your spectrum, if your spectrum is of a good quality, of course. From that particular mass, if you go back and find out how, uh, the first peak at the distance of a mass of a certain amino acid, you can be sure that that particular amino acid is at the end of that particular sequence, so on and so forth. You can keep finding out the different the differences between successive peaks. If you could define all the B and Y ions, that is a great thing. In experiments, like in experimental conditions, generally it is not possible, but you can get a combination of B and Y ions in such a way that it will cover the entire sequence of your peptide. So uh, if it is just an academic inquiry, people generally end up with a few B and few Y ions, and you can be like, it can be okay. But if it is a regulatory submission, uh, you should show the entire sequence. You should have placement of each amino acid. So maybe you'll have to optimize your methods accordingly. So what actually is done by the software is that the fragment ions will be calculated for the sequence. As I told you earlier, you should know the sequence beforehand when you are checking the quality of your product. And actually your software will prepare a list of possible B and Y ions, and it will try to match those in your fragment ion spectrum. So peptide mass fingerprinting was first developed uh, by uh, by these people, uh, Henzel et al. This is a very important paper. If any of you is more interested in the history of this particular technique, you should definitely look at this particular paper. The same paper shows you the exact thing that I was discussing, like the sequences from the database. Okay, what is the tolerance, like the error at which they are detected, what is the observed mass, the protein identified by that. Like this particular, if you are finding this set of peptides, you can be sure that this protein is present there. And like this particular spectrum will be considered as a fingerprint of your protein. So this is from the same paper. The beauty of this particular discovery or development, I won't call it discovery, it is a development. So development is that even after more than 30 years, we are still using the same basic way of dealing mass spectrometry data of a protein. So when tandem MS and database availability and computational power got added, this is again Dr. John Yates here, who designed first the approach to correlate tandem mass spectral data of peptides with their amino acid sequence in a protein database. This particular paper is a very landmark work in the field. So here is how they designed it. So they demonstrated a computer algorithm first time that converts the character-based representation of amino acid sequences in a protein database to a fragmentation pattern that can be used to match fragment ions in a tandem mass spectrometry, tandem mass spectrum. And all known sequences to correlate amino acid sequence with cDNA sequencing. So actually they harnessed the power of the genome sequencing here, and to help assign potential protein functional roles to genome sequence data. The enormous effort to sequence the human genome and the genomes of model organisms will provide complete protein complements that will facilitate the comparison and interpretation of biological studies, so on and so forth. So what they have predicted in this particular paper have actually come true. Over the years, more genomes got sequenced, and it actually prepared a basis for proteomic applications, proteomics. So here is the peptide mapping workflow. Get the expected sequence of the protein with all expected PTMs. Plan the protease, like you have to perform some in silico experiments to check which set of proteases will provide you 100% sequence coverage if you are going for a regulatory submission. Clean the sample, reduce the disulfide bonds, alkylate the thiols, digest them with proteases, the same proteases which you have designed earlier, and separate the peptides with reverse phase liquid chromatography, acquire MS and MSMS data, and match the spectra with theoretical spectra using the softwares. So it has become a very standard way of doing the things, and it establishes the primary structure of your protein, which is a very important critical quality attribute of the protein of your interest. Now coming to post-translational modifications, the same thing is used, the same workflow is used. In case of commercialized protein product, the post-translational modifications term is used to describe all the modifications that are done to the amino acid sequence. Now, if you are, adding some polyethylene glycols or some other, additional groups to your protein, that also will go to mm, uh, go under the title of post translational modification. So academically, post translational modification is something which happens inside a cell to a nascent polypeptide chain. But in case of like in scope of this particular discussion, we are using this term as a blanket term to describe all the modifications to the basic amino acid sequence. So this include all the changes, as I have said: disulfide formation, glycan, addition of glycans, addition of conjugate drugs, cutting of unwanted sequence. Sometimes, like many of the times, you will have to cut the leader and other sequences from the protein of your uh, uh, like nascent polypeptide chain. So Q6B industry guidance of US FDA asks to define the anticipated PTMs. Now there are some post-translational modifications which you are actually anticipating. You want them in your product, and there are some which appear, and then you'll have to find out like their levels and all. You know, it will be a concern to the efficacy and the it will be a quality risk, efficacy and uh, you know safety of your product will be in question. So you must be having a list of anticipated PTMs with you, irrespective of whether the PTM is anticipated or not. The testing procedure should, it remains the same, and it has to be, the modification, depending upon the modification, some customization is necessary. So in this session, I'm not including disulfide mapping, although it is a post-translational modifications. I'll just deal it separately. So these are the post-translational modifications that are generally checked. N-terminal heterogeneity, uh, modifications to the amino acids like deamidation of acidic amino acids, or isomerization of, for example, asparagine, aspartic acid can get, isomerized to isoaspartic acid. Glycation, like glycation and glycosylation are two different things chemically. Please remember that glycation is different than glycosylation. Oxidation, fragmentation, cleavage in hinge region, or disulfide bonds, free thiols, shuffling, or unwanted disulfide bond formations, thioesters, glycosylations like fucosylation, galactosylation, sialation, branching, and C-terminal heterogeneity, the lysine variants. Like there are like it again depends upon the protein you are working on. The more knowledge you have about your protein, the easier you can decide about the anticipated PTMs, and you can include them in the uh, you know list of your quality attributes. So some of these modifications like glutamate to pyroglutamate conversion, truncation of C-term lysine, they also result in charge variant of the protein of interest. Now these can be detected by ion exchange chromatography or capillary isoelectric focusing. So both can be followed by MS. Like earlier, I have described separation of peptides using or proteins using reverse phase chromatography. These chromatography techniques could be changed depending upon the nature of the post-translational modifications you're trying to identify and characterize. So here are the mass differences it makes. uh, this is not an exhaustive list, but this will be useful. This is a like these PTMs are generally uh, checked, and the monoisotopic mass changes and the average mass changes are listed here. I have also given a reference to the paper where you can find them. Of course, there is a big collection of post-translational modifications, but these are the ones which uh, mostly you would cross paths with if you're working with proteins. Now I'll come to disulfide mapping. So the correct disulfide bonding in the protein is important for its biological activity, as it provides covalent frame for the higher order structures. So that's why it is a risk with the efficacy of the product. Like if it is not correct, the product may not work as expected. uh, it if there is incorrect disulfide bonding, the folding of the protein will get affected. Some of the portions which were supposed to be hidden inside would appear to the surface of the protein, and which can cause illicit immune response. Okay, so it is also a safety risk. So the disulfide mapping is a very important attribute. If your protein uh, has one disulfide link, you will definitely have to check this particular quality attribute. There will also be a presence of free cysteines. There could also be a presence of free cysteines or free thiol groups in your protein, and this analysis is also necessary if you whenever you are working with the proteins which have cysteine residues in them. The procedure involves performing peptide mapping, the same protocol, uh, once with reduction and alkylation step and once without reduction and alkylation step, and the data from these two is actually combined to find out the disulfide map of the protein. Just a second. Okay, so here I'm showing you an example of bevacizumab. This is IgG1 molecule. Okay, so as I told you, an experiment of peptide mapping will be run twice, once with reduction in alkylation and once without reduction in alkylation. Now, and both these data will be plotted, like for a better understanding, they are plotted here as mirror plots. If you see the panel of TICs, the upper one is non-reduced with non-reduced peptide map TIC. The lower one is with reduced disulfide bonds, and there will be small differences between these two. Wherever there is a difference, it has been noted and it has been given a code, uh, you know and we will these things which are differentially appearing in both of these will actually give us the disulfide map. The non-reduced part of the TIC of the non-reduced experiment will provide you appearance of the disulfides, dipeptides which are bound together with a disulfide link, and it will have its own distinct mass which you can guess from an in silico experiment, or your software nowadays does it automatically. And the lower TIC shows, which is from the reduced experiment, it shows appearance of you know the peptides which contain cysteine and which are separated and reduced, like separated by reduction. So they will be differentially appearing here. So if you combine these two data sets, you can actually find out which particular cysteine is bound with which particular cysteine. You can find out the pairs of their bindings, and finding all the disulfide maps in your protein is mandatory. Of course, all the disulfide maps as well as your free thiols, free cysteines is mandatory for regulatory submission. Now coming to impurity profiling, purity of the product is an important aspect of every product, whether be it a protein or not. But here we are discussing a protein product. But this purity cannot be demonstrated directly. You cannot there is no test which actually shows you that the product is pure. What you have to look for is impurity. So it is always a shadow way of defining the quality of your product. So you have to decide a limit of allowed impurity, and you have to show that your like the levels of impurity in your product are below that particular level. So the Q6P specifications of USFDA actually provide a good uh, description on this. Like I have kept a small excerpt from there, like, which reads: the drug substance can include several molecular entities or variants. Now Q6B actually separates a variant from an impurity. Since proteins are complex, a single amino acid chain is not protein. You many times you have different glycoforms, variants, and they work the same. So as per Q6B, uh, there will be related substances which are product or process related, like different glycoforms or variants, charge variants, some amino acid modifications which develop over storage, like for example, you may notice increase in tryptophan oxidation over storage of your protein. So as long as their efficacy is same, they are just called as related substances. If uh, you know these entities which are different from your anticipated amino acid sequence and PTMs do not share the same efficacy, or if they pose a threat to your the safety of your product, they are called as impurities. They also could be product related or process related. They include degradation products of your protein, uh, the disulfide links which are not expected, the appearance of host cell proteins, the proteins from the host cell in which your protein of interest has been expressed, and cell culture ingredients into your product. And the third category is of contaminants. These are the bacteria, the bacterial cells which are not host, which have entered your product from somewhere outside, mycoplasma, virus, and similar things. These things, appearance of these things is called as contaminants. HRMS is useful for studying, related substances and impurities. It can also be used to study the contaminants to some extent. Now, uh, uh, to some extent, because it is difficult to know the scope of contaminants. You cannot predict beforehand which particular bacterial species has contaminated your product, or which particular virus has contaminated your product. So if you have some idea, you can always use mass spectrometry to some extent to identify the contaminants as well. So here we come to post-cell protein analysis. This is one very important critical quality attribute which is uh, addressed by high-resolution mass spectrometry. So here the workflow that is used is called as shotgun proteomics approach. This is actually proteomic identification of uh, all the proteins and peptides that are available in your sample. So here, ideally, effectively, you should be able to remove the protein, your molecule of interest, and then check what all other proteins are present in your sample. Since you know the host which you are using for expressing your protein, you can use the genome of your hostile protein as a theoretical database. uh, like from that genome, of course, you have to prepare a proteome, and you have to use that proteome as your theoretical database and match the data against it. And from this matching, you can identify the proteins. You can also identify the levels of those proteins if you further refine the technique and use some uh, I'll come to that. Low resolution techniques are available for quantitation as well. So yes, so this is about host cell protein analysis. So USP chapter 1132 describes in detail about the host cell protein analysis. It describes many different techniques, and one of them is mass spectrometry. It you know the advantage of this particular using mass spectrometry for host cell protein analysis is that it allows identification and monitoring of individual HCPs, and it can be combined with other techniques like gel electrophoresis to identify the individual proteins seen in the other techniques like gel electrophoresis. The disadvantage is low throughput, but believe me, uh, the level at which or the sensitivity at which you are able to find out the host cell proteins here, you can always uh, like the low throughput is worth going for actually. Like even though the throughput is low, you should go for mass spectrometry. So if you know the sequence, okay, 10
Excuse
nanogram
me,
per
Dr.
mg.
Hiram.
Yes.
I'm really sorry to interrupt you, but I think if possible, just try to wind up a little fast because it's quite late. Yeah, in the
Yes,
night.
yes,
So
yes, yes,
yeah. If
yes.
possible,
I'll
yeah.
sure, sure, sure. Yes. So,
Thank you. Sorry.
yes, yes, yeah. So 10 nanogram per mg is what can be achieved. Even lower can be achieved using high resolution mass spectrometry. So there is like you can if you already know what host cell proteins or other contaminants are available there, you can actually design a targeted quantization of proteins where you already I am showing here a workflow, and this particular method article I'm attributing to, you can always go to this article for more details. So the conclusion is that the CQS defined through QTPP, the quality target product profile, are a proven way of maintaining and monitoring the product quality. And in case of protein products, HRMS, allows you to do a lot many things, for checking on the quality of your product. Thank you.
So thank you, Dr. Hiram, for this wonderful talk. Okay, once again, we are really grateful to you for taking time out, again from your very busy professional and your family life, so that you can deliver the talk here. You have actually very nicely touched upon the various aspects of the has a high resolution mass spectrometry, and explain many different aspects of that. Okay, just to name a very few, like intact mass analysis, subunit mass analysis, peptide mapping, and so on. There are especially the tandem mass spectrometry, okay, peptide fingerprinting, okay, and how you can understand the post-translation modifications, impurity profiling, and so on. You have touched upon so many aspects in this, brief time. Okay, and so this, I think, gives the participants an overview of, when you are doing protein analysis. So, or by using mass spectrometry, what are the different attributes or features that you have to look at? Okay, and very interestingly, you started your talk with the aspect of that in relation to the quality. Okay, because ultimately, whatever we are trying to find out has a definite importance or bearing on the ultimate quality of your result or your analysis. So once again, we, on the behalf of the RP University, this FDP program, and the coordinators, I myself, and Dr. Moshri Madam, we are really grateful to you, okay, for coming here and delivering this really a very interesting talk. We have some questions, I guess. So with your permissions, I will just like to uh, uh, post some of them to you. Okay, so I yeah, so there is one question from Dr. Rupak Kumar. He says that, can you please elaborate about the top-down approach in brief? I think that you would have what you tried to evade has uh, come back to you. So can you just
Yeah.
briefly say about that? Yeah.
Yes. First of all, thanks everyone for patiently, going through my talk, and thanks Devashish and the management for providing me the opportunity. Now, coming to the top-down approach, top-down approach, what is done is, uh, you have your protein, you will inject it into the mass spec, and you will allow it to fragment there. You will subject it to fragmentation in your mass spec, and you will again record the fragment ion spectrum, and from those fragments, in a jigsaw puzzle way, you will rebuild the sequence of your protein. Now, this particular approach uh, is not as easy as I'm describing here, because you need a lot of pure protein. It should be very pure. You should have a mass spectrometers of certain qualities. It should cover the range. Your mass analyzer should cover the range of your the fragments that are being formed. This particular experiment requires a lot of optimization also. So that's why it is difficult to perform experiment this way. People definitely do it, uh, but a bottom up approach is easier. So that is what is used in the QC, the quality control tests. In R&D levels, when you're dealing with a certain protein with uh, you know relatively less amount of knowledge, the top down approach is advisable.
Okay. Thank you for that answer. I think we have another
uh,
same participant. Uh, I am audible to you, right? Dr.
yes,
Hiran?
yes, yes.
Okay, so Dr. Rupam Kumar is again asking that you are mentioning regulatory requirement, meaning US FDA and not about CDSCO. So he wants to confirm that which regulatory authority you are, mentioning to. It's
Yes,
US FDA
I
or
yes, I
CDSCO.
agree. So, whatever I have explained is in the light of US FDA's guidance, but uh, like I don't think in terms of spirit, all the guidelines will say the same thing. There will be some differences in the letters, or the minor details, but in spirit, I don't think there would be any difference. Even CDSCO will also require all this data. So
Yes,
it
and I just wanted to add maybe one line to your answer. Okay, because industrial experience and have been teaching these kind of subjects about quality and all in my academic career. So more or less, if we look at the the regulatory guidelines for the agencies, either it is national or international, most of it actually is same. Okay, so it's built on some kind of common guidelines. Okay, it might differ, but if you you want to talk on a percentage level, I think at least 70 to 80% are same, because the quality attributes of any work or any particular experiment or the results remain same globally. Okay, so it does not actually change much, from one guideline to guideline, is it not? Yeah.
I agree with Devashish.
Yes. So I think if we are not having any more questions, we can, conclude this session now. And anyway, the email IDs of both these respected speakers are shared in the chat. So all of you can, if you have any further questions on the sessions today, you can directly contact them. Okay, I think they'll be very happy to answer. So, Dr. Hiram, once again, we are really grateful to you. Thank you so much for coming here and delivering this talk. We hope to extend this collaboration with you even in future. Okay, thank you so much.
Thank you, sir.
Partha,
Thank you so much.
can we have a good round of applause for Dr. Hiram? अच्छा round of applause हो जाए. Okay, so now I like to hand over this session to my respected co-coordinator, Mushrini Madam, to please conclude the sessions today.
So we are at the end of the session, and we are now winding up tonight's session with the two brilliant speakers. One is Dr. Shoptak Banerjee, and another one is right now we have finished, Dr. Hiram Kulkarni. And I guess the speakers still now present might have really experienced a good exposures for oncology or cancer biology, and also the structural analysis of the proteins by high resolution mass spectrometry, and as well as tandem MS mass spec analysis, and all the advanced things which we generally won't come across in usual academic or research life very frequently. So with this short note, I thank both the speakers from bottom of my heart with my co-coordinator Dr. Devashish Banerjee, and on behalf of Atmiya University Rajkot and all the management team and Atal AICT funding support agency. It's our great privilege to have both of you in this particular platform in a single roof. Good night, all of you.
Before just signing off, we can ask the participants to kindly click on the
I
video.
guess if they are agree, then we can have a group photo, because both the
Yes.
speakers are there, and as well as if they are not so tired, then we can request just
whoever
we can see the encouragement by the instant response, and it is really making us more and more every day to do more improvisations. Thank you so much.
पार्थ भाई आप काइंडली ये लेते रहिए जो snapshots sir. Thank you so much all the participants for
Thank you
your
so much
encouraged response.
for the response. Thank you so much.
Okay, I think that's enough. So
we can wind up the sessions, and tomorrow
yes.
at the same time, 6 o'clock, we are again joining with again two brilliant speakers, two marvelous topics. We are looking forward for your participation tomorrow. And once again, happy Makar Sankranti, happy Lohri, and happy Uttarayan for all of the Indians from our sides. Enjoy the festivity, and along with that, please join us in that festive mood tomorrow. Thank you so much. Good night, sir. Good night, sir. Thank you so much. We are winding up here.
Thank you, madam. See you all tomorrow. Thank you once again. Goodbye.
Thank you. Thank you so much. Goodbye.